All right, good afternoon. Today is Tuesday, September 29th, 2020. We are going to begin our long and arduous journey through the endocrine system, talking about everything you ever wanted to know and more about the endocrine system over the next couple of days. So again, it's going to be long, boring, exhausting material, but we will do the best that we can, talking about the material and the histology for it to make ourselves comfortable with this material as much as we can. You have really two more assignments due. One is the Labster, which is going to be due on Thursday. Remember, you must complete it 100% completed and 80% correct for full credit. Again, remember when you complete these Labsters, because it's synced with Canvas, the grade automatically gets input. So if you got 95%, it gives you 95% of the points. But remember, as long as you get at least 80% correct, I will go in there and correct and change the grade manually to 20 points, full points. However, if you get less than 80 points, 80%, you will get fewer points. So it is important to remember you can do this as many times as you want. So make sure you complete the whole thing and get 80% correct for full credit on that. Then, and again, I made it bright and bold. I don't know how many times I can warn you guys about this to make sure it gets done. But remember, again, most of the assignments are due on the day when we have class, and that is important, I think, especially in a synchronous class like this. But to help everybody to prepare for Tuesday's group presentations, everybody's outlines are due on Monday by 5 p.m. So again, just one outline per group. Take all of your material, condense it down into one presentation, condense it down into one outline. That outline should have everyone's name on it. So again, let me write this to remind everybody. Have everyone's name, full name. I think it helps the class to get to know everybody, and also the name of your disorder on it. Please do not forget to put that on there as well. And they're due in the Dropbox by 5 p.m. on Monday. Again, if you do not turn that in, then it's either late or missing. You will lose 10 points on your presentation. Your presentation is 50 points, so that is a huge chunk. So it is important to make sure you turn that in on time so that I can post those onto Canvas so people have the opportunity to take a look at them ahead of time, print them out if they want to ahead of time, to make sure that they have this material to help them to be successful studying this material for the exam that you're going to be presenting. And again, the outline should be an outline. It should not be a script of the material. It shouldn't be everything that everybody needs, but you want to give them those breadcrumbs to help them follow along as you give your presentation. And that's what we'll be doing, hopefully, all day on Tuesday. If we run a little behind on the endocrine system, we may have to lecture a little bit at the beginning. I'm hoping for that not to be the case. And so today's and Thursday's lectures are going to be longer to make sure that we have time to be able to just do group presentations on Tuesday. And then Thursday, you have the lab and lecture exams, the exact same format as the first one. All right, and I'll remind you of all the rules and everything of that as we get closer to it.
Will we do any review on Tuesday?
I'm sorry, say again?
Will we do any review on Tuesday?
If we get the presentations done in time, and I think we have 11 groups. Yeah, we have 11 groups. So usually it's about 3 groups an hour. So if that's the case, then we should be done in less than 4 hours. And then, yes, even with breaks and stuff. And then, so yes, any time that remains would be an opportunity for question and answer review, absolutely. All righty. Any other questions? All right, excellent. Then let's get started. Our job now is to discuss the endocrine system. All right. As we learned in 430, there's a big difference between endocrine glands and exocrine glands. What does exocrine mean again? Well, what does exo mean?
Outside.
Outside. What does crine mean? Anyone know?
Inside.
No. Crine means to secrete.
Sorry.
Yeah. So exocrine glands secrete outside the body. And what is one of the key characteristics of our exocrine glands that allows them to secrete, release their material outside of the body? They have ducts. Remember when we learned about our exocrine glands in 430, we learned about, tubular, alveolar, right? Simple, branched, compound, all of those terms sound vaguely familiar. And all of those exocrine glands had both secretory structures and ducts because their job was to secrete outside of the body. Endocrine glands, on the other side, crine still means to secrete. What does endo mean?
Inside.
Yeah, within, exactly. So endocrine glands release their contents inside of the body. Instead, they release their contents into the humors of the body. Right? Primarily, what humor do they release their contents into?
Lymph.
When it releases it into a fluid of the body, you're right in that it is going to end up into the blood, but most of them don't actually directly release into the blood. Where do they directly release into? There you go, interstitial fluid.
Fluid? Oh.
Yeah, into the interstitial fluid, but then you guys are absolutely correct. Once it gets into the interstitial fluid, it ends up in the blood, and once it ends up in the blood, where does it go?
Lymph.
Well, it can go into the lymph, but also when it gets in the blood, what parts of the body can go to? Everywhere. Right? Some of these circulating hormones that we release have the potential to affect every single cell in the body. Now, is every single hormone going to affect every single cell in the body?
No.
No, of course not. But by getting into the humors of the body, by getting into the blood, it has the potential to go everywhere and affect everything. Right? Now, since they are just releasing this into the humors of the body, our endocrine glands are ductless, right? They do not have ducts. I don't know if ductless is a word, but I'm making it one. So they're ductless. And of course, what is the substances that we call, what do we call the substances that endocrine glands release? Hormones, absolutely. They produce and release hormones. All righty. Excellent. Now, the other thing that when we talk about our endocrine glands, we've talked about relating our endocrine glands to our exocrine glands, but the other thing we related our endocrine system to, or endocrine glands to, is the nervous system. Why are these two often compared with each other? What's special about both these systems? True, absolutely, right? They produce signals, and really, well, the way I like to say it, and again, saying that they produce signals is perfectly acceptable. Both are specialized for communication. Right? That is a characteristic that both of these organ systems have. Of course, how does our nervous system communicate? What is its signal?
All impulses?
Sure, absolutely. So impulses, electrical impulses. Do we have a special name for the big electrical impulse that we use to send signals long distances? Well, I'm asking the question, so clearly we do.
Action potentials.
Action potentials, excellent. You are correct in that it is the neurons that produce the action potentials. But those action potentials are those big positive signals that are produced by our neurons in our nervous system. Whereas the endocrine system, as we already talked about, is a chemical signal that we call a hormone. While both these systems are specialized for communication, what's the big difference between them? Speed, absolutely. Right? Electrical impulses are fast, whereas producing a chemical signal, think about this. If you put your hand on something hot, when that tissue is damaged, would you want that tissue to have to go through the process of making a protein where we have to read the DNA, and then we make a messenger RNA, and we send that messenger RNA to the rough endoplasmic reticulum, where translation takes place to produce a sequence of amino acids, which then goes to the Golgi apparatus to be cut up, folded, modified, bundled up in a vesicle with enough of them that they can be brought to the plasma membrane, released into the interstitial fluid, get into the blood, circulate till it gets to the brain, and then the brain can do the same thing, make a messenger RNA, make a protein, release it into the interstitial fluid, get into the blood, circulate to the body, finally get back to the hand and tell you to move your hand out of the way? No, your hand would be burnt off by the time that was done, right? The advantage of the nervous system is fast, but it is also a transient system, or a transient signal. It is very, very rapid, like waves in a pool. Remember, we talked a lot about waves in a pool. But even if I take a massive rock and throw it in a pool and produce a massive wave, how long does that wave last for? Not very long. Right? So whereas our chemical signals is a slower signal, it is also a longer lasting signal. So this allows us to coordinate information and coordinate processes over hours, days, weeks, even years, right? How many of you are the exact same size now as you were when you were born? Anyone?
Hope not.
Yeah, I hope not for your mom's sake. I hope not as well. Absolutely, right? So obviously going from a tiny baby to the big, massive, mature adult that you are now took years to accomplish and required the coordination of processes in your body done by the chemical signals of our endocrine system, right? And so that is our job, to produce these signals, to produce those hormones, and to spread them to coordinate that long-lasting signal throughout the body. And of course, the study of the endocrine system is the field of endocrinology. Let's talk more about these general functions. Again, it is specialized for communication. So we have that ability to be able to coordinate the activity of most of the cells of the body. So it plays an important role in our metabolism, regulating our biological clock, our intracellular fluid levels, helping us to deal with stressors. All of these are things that our endocrine system helps us to do. And then, like I said, it helps to coordinate all of these long-lasting processes like growth, like development, right? So not only do we go from babies to adults, but we go from boys to men. We go from girls to women. And then obviously, the endocrine system plays a very important role in producing other offspring. The maturation, the development, and the maintenance of our reproductive system, right? Making those puzzle pieces, putting those puzzle pieces together, housing them to make new people. And again, really, this is just the tip of the iceberg of the stuff that our endocrine system and hormones do for us. All right, questions on that? All right, as we've talked about, one of the keys with our endocrine system is our endocrine glands are typically a cell or a cluster of cells that are located in just one region. Then their job is to produce and release its contents, like we said, into the interstitial fluid. But once it gets into the interstitial fluid, it can get into the blood. And once it gets to the blood, it can travel anywhere to the body with the potential to affect any cell in the body. But as we talked about, it isn't going to affect every cell. A hormone can only target a cell that has a specific binding site for that hormone. And those cells that have the binding site for that hormone, we call our target cells.
Those binding sites, it's receptors, right?
Yes, absolutely. The binding sites are a receptor. So we have a receptor, and that receptor, as we'll talk about, can either be on the surface of the cell like these are, or actually sometimes they can be inside the cell as well. All right. And here's an example of this. Hormones can only affect specific cells that have a specific hormone receptor. So each receptor is specific for each hormone. So if we cheat and draw a cell, here's my circle. Here's a cell. That cell will have a binding site on its surface. And what can happen is that our hormone can come in, and I know I didn't line that up properly, but you get the idea. Come in and bind to that receptor. And when it binds to that receptor, it forms what we call the, not surprisingly, hormone receptor complex. And really, it is this hormone receptor complex that is going to affect the change inside of the cell. So it's the hormone receptor complex, the two things together, both the hormone and the receptor, that affect the change in the cell. All right. Now again, let me clear my drawing because I've got a better one. So notice here we have two different hormones. Hormone A, which can combine to this cell. Hormone A, which combine to this cell. Hormone B, which combine to this cell. Hormone B, which combine to this cell. So notice this yellow cell is only a target cell for hormone A. This green cell is only a target cell for hormone B, whereas the blue cell is a target cell for both A and B. No, great question. So I know my drawing may have looked a little bit like a major histocompatibility complex, but no, that's not. And I notice these things kind of look a little bit like antibodies. No, these are, here's a great term. These are going to be just receptors, hormone receptors. Some of them, and we'll talk about this more in just a minute, but some will be transmembrane proteins. What does that mean to be a transmembrane protein?
It transfers inside of the cell.
Across the plasma membrane.
Careful, because when you say that it transfers inside of the cell, that reminds me a little bit of more like a channel or a carrier, where it allows the hormone to get into the cell. That isn't necessarily what's happening here. What does it mean to be a transmembrane protein? Multiple membranes is closer, but if only I remembered what my phospholipid bilayer looked like. Right? My plasma membrane is that phospholipid bilayer with two head regions, and then the tails, tail, 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 tail. And with that phospholipid bilayer, you have the heads out and the tails in, right? That's what our plasma membrane looks like. A transmembrane protein is an integral membrane protein, meaning that it is a protein that is embedded within the plasma membrane. But a transmembrane protein goes through both layers, right? It's possible to have a protein that is just embedded in one layer of the phospholipid bilayer, and that would still be a integral membrane protein. But a transmembrane protein is an integral membrane protein that goes through both layers of the phospholipid bilayer, so that a hormone can bind to it on the outside, but it can affect some type of change on the inside. But some receptors can actually be found inside the cell. Yeah, let's continue the discussion. We'll see more examples of this. Now, one of the cool things about this is a constantly dynamic, constantly changing process where there is up and down regulation, right? So again, the example I like to think of it is that as a brand new parent of a newborn, especially your first child, right? That in the case, you are going to have a massive number of receptors on the outer surface, because if the baby even just whimpers a little bit, it gets your undivided attention and you are instantly responding to it. So if I have a cell that has 1 million receptors on the surface, and I sprinkle even just the tiniest bit, right? 4 tiny little neurotransmitters get released. 4 tiny little hormones get released. If this cell has 1 million receptors for it, Is it likely that one of these hormones is going to bind to one of these receptors and have some effect on the inside of the cell? Yeah, absolutely. Right? But then your kid becomes a teenager. And as the kid becomes the teenager, you become less sensitive to all the noise that they make, to the point where they could be standing right next to you, screaming in your ear, and you're hardly aware that they're there at all. If the cell only has one receptor, and I dump a whole bucket of hormone over the top of this cell, is it going to have a big effect on this cell? Now, not if it only has one receptor. It's likely not to have a big effect. And let's go to the extreme. What if I remove all of the receptors? Now, if I dump 2 buckets of hormones on this cell, is it going to have any effect at all?
No.
No. In fact, at this point, the cell would be insensitive. It would be insensitive to the hormone. Conversely, by gene expression, I could express a brand new protein on the surface of the cell that was never there before, and now suddenly this cell becomes sensitive to a hormone that it wasn't sensitive to before. So really, when we're talking about this up and down regulation of receptors, since receptors are protein, what we're really talking about is a change in the expression of proteins for the cell. We can make more of the receptors, we can make less of the receptors, and that will make the cell more or less sensitive to the hormone. Does that make sense? Excellent. My blank and utter silence means that I totally understand this, and we move on. All right, excellent. Now, again, when we talk about our hormones, especially up to this point in time, we've primarily been talking about circulating hormones. Circulating hormones do exactly what we were just talking about. One cell or cluster of cells in one location releases that long-lasting, stable hormone into the interstitial fluid, where it then gets into the blood and circulates throughout the body. And that's the key. This is a long-lasting chemical signal. And it's able to be a long-lasting chemical signal because typically it is a stable protein. Let's just say molecule. It is a stable molecule. So it's a long-lasting, stable molecule, which allows it to be circulated throughout the body. However, one of the things that we started to learn about when we were talking about our immune response is we were talking about things like those cytokines. And remember with the cytokines aren't about getting into the body and going everywhere, but having a local effect. Interferons were chemical signals with a local effect. Some hormones can be local hormones. These local hormones are less stable, less long-lasting. So they can't travel far different distances. But what it allows them to have are local effects. One example of this is what we would call a paracrine signal. Paracrine is where one cell releases a chemical signal that affects the nearby target cells. And the other type is an autocrine. An autocrine is when a cell releases a chemical signal, a hormone, that actually stimulates itself. We actually talked about a cell that released an autocrine hormone signal. What was the cell that we talked about that when activated, one of the first things it does is stimulate itself? What were those cells?
Smooth muscle cells.
No, not smooth muscle.
The helper T cell?
There you go, the helper T cells. Remember, when the helper T cells are activated and they start releasing those cytokines, one of the first things that we said they do is they actually stimulate the division of more helper T cells. That would be an example of an autocrine signal. However, when those helper T cells release the signal that activates the B cells, so the B cells divide, that would be an example of a paracrine type of signal. So we have actually talked about these types of chemical signals before. We just haven't used these fancy terms for them yet. So it's important to know that not all hormones are circulating hormones. There are some local hormones as well, and at least one on your list that you're responsible for. Let's see if you guys can figure out which one that is. Stability, by stability, what I mean is how long the molecule lasts. Remember, all of these chemicals are released into the interstitial fluid, into the blood, right? And they're like Legos. If you build something, a really huge, long line of Legos that are simply made up of a single cubes, right? A whole long line of simple cubes. Is that going to have a lot of stability? Am I going to be able to have, my kids going to be able to have sword fights with Legos that have just been a single layer, a single square box, all put together into a row? No, it's not a stable structure. It's not long lasting. So as these things bounce around and bump around inside the blood vessels or around other things in interstitial fluid, they break down, they degrade, they get broken down by enzymes. So they're not long lasting. They're not very stable molecules. Whereas other ones have more structure to them. When they have the more structure to them, they're less likely to break down. They're more longer lasting. Does that answer that question? All right, excellent. Perfect. All righty. So, when talking about hormones in general, and then we'll get into more specifics in a moment, as we mentioned, our hormones have to bind to a target receptor, forming that hormone receptor complex. I have now used that term twice and probably will at least 2 or 3 more times. So this would be the point where you're guessing that's an important phrase that you need to know. And as I mentioned, it can happen both inside or on the plasma membrane. If it happens on the plasma membrane, if you think about it, the hormone does not have to enter the cell. Whereas if the hormone receptor complex is going to take place inside the cell, in that case, the hormone must enter the cell. So where it binds to the receptor is either going to be outside of the cell or inside of the cell. When a hormone binds to a cell, binds to that receptor, forms that hormone receptor complex, basically one of several things are going to occur. The cell can start to make new molecules, primarily new proteins. Right? Gene expression, right? Leading to transcription, leading to translation, leading to the making of new proteins and the expressing of those proteins or the releasing of those proteins. When we do that, we can change the permeability of the membrane by adding or removing channels, carriers, and pumps to the surface of a plasma membrane. We can change what gets into the cell and what gets out of the cell. So we can change its permeability. And that can change the work that it's able to do. And along those lines, we can also affect and modify the metabolism. If you think about it, again, metabolism is the total number of chemical reactions. That occur inside of a cell. So, for instance, we can turn enzymes on, or we could turn enzymes off. If we turn enzymes on, what happens to the number of chemical reactions that take place during a period of time? Increases, and so metabolism is increased. Conversely, if we turn enzymes off, number of chemical reactions go down and metabolism goes down. So in this way, we can change the behavior of the cell in one or more of these 3 ways. One of the challenges to, I'm sorry, was it a question? Background noise, okay. One of the challenges of studying the endocrine system, and one of the reasons why it's one of the studies, the areas we still know probably the least amount about, is because the interactions between hormones and cells can vary differently, right? Each target cell can respond differently to the exact same hormone. Let's talk about a really simple example. We in the last class talked about insulin, and we talked about how insulin plays an important role in regulating blood glucose levels. One of the things insulin tells, we said insulin affects most of the cells in the body, but one of the ones we said it particularly affects are liver cells. And it tells those liver cells to take glucose out of the blood and store that glucose inside of them. And if liver is going to store glucose, what does it store that glucose as?
Fat?
Well, does it store it as fat in the liver cells?
Glycogen, yeah.
Yeah, glycogen, big, huge glycogen molecules, absolutely. However, as someone just mentioned, insulin also can affect adipocytes. And if it affects adipocytes, those adipocytes will pull glucose out of the blood plasma, out of the interstitial fluid as well. And when an adipose stores that glucose, what form does it store it as?
Fats.
Right, or specifically triglycerides, right? And so notice insulin tells liver to grab glucose and make glycogen, whereas insulin tells adipocytes, grab glucose and make triglycerides. So the problem is every single target cell to a hormone can have a different effect from that hormone. So there isn't just one effect for insulin. Insulin has many effects. And so do other hormones as well. So that's one of the things that we're going to talk about and need to work on as we're talking about the targets for particular hormones. We're going to have to make sure we understand what the effects are. If something has 3 different targets, we need to make sure we understand what the different effects are on those 3 different targets. All right? Questions on that? All right.
Insulin also allows this glucose to enter body cells, right?
True, absolutely. And so you're correct. As we mentioned, insulin tells most of the cells of the body to absorb glucose. And most of the cells of the body that absorb those glucose, how do most of the cells of the body store that glucose?
As fat, right?
Say again?
As fat?
No, most of the cells actually stored in the form of glycogen. Right? Most of them are stored as glycogen. Glycogen is stored in the cells, but adipocytes store it as fats. So again, there's a great question here. Remember, an adipocyte is a cell. So it is a cell that has a nucleus, has organelles, has all of those things inside of it. But it is a specialized cell that inside of it contains a specialized organelle. That specialized organelle is called a fat vacuole. And that fat vacuole basically is a big, huge plasma membrane that stores triglycerides. Remember, triglycerides are lipids. They are fats. It is that neutral fat with a single glycerol and 3 fatty acids that connect to it. And so those triglycerides are stored as an oil droplet inside of the cell. Remember, that tends to shove the nucleus of the cell way to the side. So it kind of gives our adipocytes that kind of diamond ring appearance. So an adipocyte is a cell. Triglycerides, remember, are a macromolecule or a lipid. And so again, if you need help with the macromolecules, chapter 2, remember, is your chemistry material. So make sure you're familiar with those macromolecules like proteins and things along those lines. Oh, okay. Great question. So again, this one was sent privately to me, so I'll repeat the question. The question was, is there a way to specifically target adipocytes for apoptosis? So if you get them to destroy themselves, then suddenly that would be a great way to lose the weight. The problem with that is twofold. The first is that if the cell dies, it isn't like the contents of it disappear. What you would be doing instead is releasing all of those fats inside of it into the interstitial fluid, which could then get into the blood. So you would have this massive release of triglycerides into the blood, which would be very, very dangerous for that to happen that way. And it doesn't get it out of the body. Now, what we did want to do is, luckily they have vacuums that they could stick underneath your skin and suck all those adipocytes out. What do they call that process?
Liposuction.
Liposuction, which is an awesome way to lose the weight, because one of the things that I'm sure you remember from 430 is that adipocytes don't divide. What happens is you have this one adipocyte here. If the amount of calories that you take in is greater than the calories you expend, then it accumulates triglycerides in it and the cell gets bigger. However, if you ingest fewer calories than you expend, then we have to get these resources from here and the cell shrinks. It never goes away. So we can make the cells bigger as we gain weight, smaller as we lose weight. But we don't want to put in all the effort and time. We can just use that vacuum to suck them all out. And lo and behold, I can go back to eating 17 cheeseburgers for breakfast, 17 more for lunch, and 17 more for dinner, because I've sucked all the adipocytes out of my body. And adipocytes, there may be a couple left that remain, but they can't divide. So I will now stay thin forever, yes?
No.
No.
Why not?
What about stem cells?
Ah, exactly. If only there was a pluripotent stem cell that was capable of dividing to produce all of the cells associated with my connective tissues. Does something like that exist? Of course it does. What is it? What is the name of the pluripotent stem cell that is capable of dividing to produce all of the cells associated with our connective tissues? Adipocytes, chondrocytes, osteocytes, fibroblasts, everything. Anyone remember? Mesenchymal stem cells. Excellent. So yes, so no, getting them to undergo apoptosis wouldn't help in that process. Great question though. Any others?
Yeah, I have a question about, so if you get a blood test and your triglyceride levels are high, does that mean that the triglycerides are just remaining in your blood because your adipocytes are full? Or is that just a measure of how much triglycerides you have in your adipocytes?
Great question. It is not a measure. So one of the things that I think one of the misunderstandings about our adipocytes is our adipocytes are incredibly dynamic. They're constantly breaking down those that fat vacuoles into basically lipoproteins, basically lipids surrounded by proteins that can then be transmitted throughout the body. So they're constantly absorbing new and releasing new. And so there is this constant dynamic spread of lipids throughout the body. We do it bound to polar proteins, so it's not congesting the blood. But the more adipocytes to have, the more fat there is in your diet, the more will be transmitted through the blood. So yeah, so it is a measure of how much is in the blood, but the stuff that are in the blood is bound to lipid-soluble proteins. That is how it's transmitted. So it's not like once it gets into the adipocyte, it's locked in there in a locker and never leaves. It's constantly being rotated throughout the body. All right?
Okay, so if you have too much of it, it just constantly is being rotated.
Yes. Yeah, and then it gets stored in places like your liver. So your liver can become very fatty as a result of it, places like that. So yeah, it will start to accumulate in places, the pericardium, your mesenteries in the digestive system, lots of places where it will start to accumulate, yes. And there will be excessive amounts in the blood, which is bad. All right? All right. So, as I mentioned, it is both the hormone and the receptor together, that complex that determines the effect. All right? So as we talked about, oops, hold on. I have a cell like that liver cell with a receptor on its surface. And hormone, like insulin, can come and bind to that hormone. And when it binds here, let's actually put it in here a little nicer. There you go, something like that. And then I'll erase the first one. When it binds to that receptor, that hormone receptor complex is going to cause and infect some kind of cell. So as we talked about, it will tell this cell to grab and store glucose. All right? That is the function of that hormone on this cell by itself. And the more receptors or the more hormones it has, the more of this it will do. All right? So far, so good?
Yes.
Part of the problem with hormones is that different hormones can have different effects and interact with other hormones. So let's switch to the whiteboard. I think this will be easier to show in a really simple illustration. All right, here we have hormone 1. Actually, I lied. Here we have hormone 1. Right? Hormone 1 is our little triangle. And let's say, for argument's sake, that hormone 1 doubles the metabolism of the target cell. Okay? So far, so good? Here, hold on. I need to do that. Oh, this will work. I like this. I need this out of my way. I need that out of my way. Okay, perfect. Hormone 1 doubles the metabolism of the target cell. All right. So here we have the target cell. And so our target cell is here. And this target cell obviously is a target cell, so it has a binding site for hormone 1. And we'll make hormone 1 a red triangle. So when hormone 1 binds to the triangle, it doubles its metabolism. Make sense? Everybody comfortable with that?
Yes.
Sounds excellent. However, what's interesting is this cell isn't just a target cell for hormone 1. There can be a second hormone, second hormone receptor, and we'll make this one a circle, for hormone 2. And hormone 2 can modify the effect of hormone 1. And there are 3 different ways that hormone 2 can modify the effect of hormone 1. So notice what is happening here is we are getting hormone interactions. There are 3 hormone interactions. Where there's 3 different ways that hormone 2, my little blue circle hormone, when it binds to the target cell, can change the overall effect. All right. Do we get how the game is played? All right. What does hormone 1 do to the target cell?
Doubles metabolism.
Excellent. One possible interaction that can occur, so here, let's do this. So this is the setting of the table. We know hormone 1 always doubles the metabolism, so that makes it easy. It always is going to double the metabolism. But what can happen is that hormone 2 may also increase the metabolism. And let's say it also doubles the metabolism. So hormone 1 doubles the metabolism. Hormone 2 doubles the metabolism. So if both by themselves, let's emphasize that, right? Doubles the metabolism by itself. But when the 2 are together, so the overall effect, what would you expect the overall effect to be? If hormone 1 doubles the metabolism and hormone 2 doubles the metabolism, what would you think the effect of both of it would be? It would be 4 times the metabolism, right? But it's not. It's 10 times the metabolism. Is that additive? Normally, we would expect these effects to be additive, but it's not. And again, I just made up the number 10. But the key to this type of interaction is we get an amplification of the enhancement, like 10 times the metabolism. It's not additive. So we are getting an amplification of the enhancement. It's not just adding the 2 of those together. And we get this big, huge effect. All right. This type of interaction we call a synergistic interaction, where you get an amplification of the effect. Questions on that? Well, there are two other ways that we can have an interaction. So let's see. Yes, all synergistic interactions involve amplification. Yes. And as Riley pointed out, absolutely the opposite can happen as well. Let's say this one doubles the metabolism. Right? We know hormone 1 always doubles the metabolism. That effect doesn't change. But hormone 2 by itself reduces the metabolism. And we don't have to bother putting numbers with these because we're just making up the numbers anyway. So when you put the 2 of these together, what effect does that have on hormone 1's influence? Are we still going to be able to double the metabolism of this target cell?
No.
No, instead of amplifying the effect, it is going to reduce the effect of hormone 1. And is it possible it could even cancel it out?
Yes.
Yeah.
So that there's no effect. Excellent. So absolutely, it could reduce the effect, and that, not surprisingly, we call antagonistic. Absolutely. I think those are the two easy ones. So that's the ones I started with first. Because this last one is where it gets a tiny bit tricky. So I want to make sure we understand the basics of this here. Notice what we are looking at is the effect of these two hormones together. Hormone 2 present at the same time as hormone 1 changes the effect of hormone 1. Okay? So let's look at our last case. We know that hormone 1 always doubles the metabolism of the cell. So there I am in my lab studying this. I sprinkle hormone 1 on the target cell and see what it does. It doubles the metabolism. Then I sprinkle hormone 2 on the cell. And when I sprinkle hormone 2 on the cell, by itself, the hormone does nothing. The hormone has no effect by itself. Well, if you think about it, if you're in a lab and you have a cell and you sprinkle a hormone on it and nothing happens to the cell, you throw away that hormone, right? Done a study. I know this hormone, hormone 2, doesn't affect the target cell, so I don't have to worry about studying anymore. So I can just throw away my whole bag of hormone 2 because I don't need it. But then I have some pesky grad students, and grad students are always messing things up. And that grad student accidentally puts 1 and 2 on the target cell at the same time. And when that happens, what occurs is, once again, we get an amplification of the effect of hormone 1. The effect is greater than just hormone 1 by itself. So again, it could be another case where we have a 10x increase suddenly.
I don't understand why.
The reason why is because hormone 2 has what we call a permissive interaction. Hormone 2 doesn't do anything by itself, but what it does do is enhance hormone 1's effect.
Oh, okay.
All right? If your job was to get over a fence, you could stand at the bottom of the fence and you could climb over that fence by climbing up the fence, swinging your legs over the top, and jumping over. And if there was a party of 10 of you and you all had to get over the fence, right, it would take you guys 10, 20 minutes for everybody to be able to do that. But what if we put a ladder up against the fence? If you put a ladder up against the fence and you just leave it there, nothing happens. But when you have 10 people trying to get over that fence, that ladder makes it a lot easier. And you guys would get over a lot faster. And that's what's happening here. Hormone 2 does not have an effect by itself, but it enhances the effect of hormone 1. And that's part of the problems. When you first start studying hormones, when you pour a hormone on a target cell and it does nothing, does that mean you can stop studying that hormone? No, because now you need to see how it interacts with every other hormone that affects it as well. It doesn't help 2, 2 helps 1. Hormone 2 is the latter. It helps 1 do its job and does its job more efficiently. So notice, basically synergistic and permissive, you get the same effect. So notice in both of these, you are getting the same ultimate effect. When both hormones are present at the same time, you get the same effect. The big difference between synergistic and permissive is by itself, hormone 2 has an effect with synergistic. By itself, hormone 2 does nothing with permissive.
But the effect the same.
The overall effect at the end is the same. Both amplify the effect of hormone 1. We get an enhancement of the effect. So the overall, the end result is the same. The big difference is what hormone 2 does by itself.
So if synergistic and antagonistic were just by themselves, they would do that. They would do their own effects. But if
Yes.
permissive was there by itself, it wouldn't do anything, basically.
Correct. So yes. So notice what we have in this column is the effect of hormone B, hormone 2. Hormone 2 by itself here enhances the metabolism. Hormone 2 by itself reduces the metabolism. Here, hormone 2 by itself does nothing. Yes, our body is indeed so weird. Oh, these are just even the chemicals inside of our body. So yeah. All right, so let's do that. Go back to the pretty picture from your textbook. And like we said, in a synergistic effect, the two hormones have the same effect. So again, the key here, they have the same effect. Or same type of effect, let's say that. By themselves, and enhance the effect together. That is our synergistic effect. So the two hormones are working together, doing the same thing, and together they enhance and more specifically amplify the effect together. Permissive also amplifies the effect. But in this case, hormone 2 has no effect by itself. So the hormone 2 is just helping hormone 1 do its job. And then, right, antagonistic. Antagonistic is something that we're familiar with from, right, studying the studying the autonomic nervous system, right? We'd always talked about how sympathetic and parasympathetic are antagonistic. If one increases heart rate, the other decreases the heart rate, right? If one increases digestion, the other decreases digestion. So here we have two hormones having opposite effects, and they are going to reduce the effect and could even cancel out. These are our interactions. All right, interactions. I make a point of emphasizing this because I'm also going to ask you about hormonal regulations. And we actually talked about that at the end of last class. Anybody remember there are 3 interactions. There are also 3 ways of regulating the hormones. Anybody remember what the 3 ways we talked about regulating the hormones would be? Hormonal, neural, and humoral. Excellent. Those are regulations. So the same way on a lab exam, if I asked you for the contractile cells and you gave me the autorhythmic cells, you get that question wrong. Same thing here. If I ask you for hormonal interactions and you give me regulations, you're going to get that wrong. So read the questions carefully so you can answer them correctly. So we have talked about interactions. Let's now talk about hormone regulations, how we regulate how hormones are released. Now, we already mentioned the 3 regulations of how we can modify it, but what we didn't talk about is how is most hormone levels maintained in the body? How do I maintain an appropriate amount of insulin in my body?
Increasing or decreasing is in blood.
Yeah, how would I do that? Or why would I do that? How would I know to increase it or decrease it in the blood? There you go, absolutely. The primary way we're going to do this is absolutely using negative feedback mechanisms. Our goal is to have 10 microliters of insulin in our blood, or I should say per micro. I agree. Actually, micro units is what it would be. Units per microliter of blood or something like that. Again, I'm totally making this up, so. Right, or something like that, whatever that number is, right? And as our insulin level drops, we bring it back up. If insulin goes high, we stop releasing it so that it'll come down. The primary way we control the levels of hormones in our body is with negative feedback mechanisms. Not every hormone, but most. This is going to be, so it's the primary way we control most. But insulin's a great example. You want a relatively stable amount of insulin in your body until you go to that birthday party, right? Back in ancient times, we used to have birthday parties, not birthday parades. And when you would get to the birthday party, among the other things you would get is a slice of very carb-rich pizza, followed by a large chunk of cake, 2 scoops of ice cream, and then a bag of candy to take home and eat in the car, right? And what happens to your blood glucose levels at the end of that party? Yeah, they're through the roofs. And if they're through the roofs, do we want to maintain that same amount of insulin? No, we want to be able to modify. So while primarily we use negative feedback to maintain normal levels, right? This is our job, to maintain normal levels under normal conditions. There are times when we need to change it. There are going to be times when, because of circumstances, like that birthday party, and if we want to be more fancy, we can say because of homeostatic imbalances, that we want to modify it. And if we're going to modify the levels, there are 1 of 3 ways to regulate that hormone level. All right, oops, and now that I've put that there, I don't have anywhere else to put it, I'm just gonna go ahead and clear it. We've talked about what they are. Hormonal control. Hormonal control is where one hormone controls the release of a second hormone. We see an example of this here with our hypothalamus and our pituitary gland. Our pituitary gland produces and releases about a dozen hormones, but let's start easy. One of the hormones that is produced and released in our anterior pituitary is thyroid stimulating hormone. Whoops. All right. This is one of the hormones that is present and produced and released by our anterior pituitary. What our hypothalamus does is it produces a hormone called, and I'm going to abbreviate here, thyroid stimulating hormone releasing hormone. So the hypothalamus produces a hormone. This is hormone A. And hormone A tells the anterior pituitary to produce and release more of hormone B. thyroid stimulating hormone. So notice one hormone produced in one location tells a different gland to produce more of its hormone. And it gets even more fun than that. Where do you think thyroid stimulating hormone goes?
Thyroid gland.
Goes to the thyroid gland. What do you think it tells the thyroid gland to do?
To produce thyroxine.
There you go, and triiodothyronine, right? So it's going to affect what we call the thyroid, the class, thyroid hormones. And those thyroid hormones for now, for space, T3 and T4. Thyroxine, well, triiodothyronine and thyroxine. And so once again, we have a hormone telling a second gland, a second hormone, hormone C, to be produced. So notice here, we actually have two different levels of hormonal control. Thyroid stimulating hormone is controlled hormonally. Thyroid hormones are controlled hormonally. And in fact, I think the pretty picture shows us that. Thyroid stimulating hormone stimulates the thyroid to make its gland. There are hormones that tell the cortex of the adrenal gland to release its hormones. We have our gonadotropins that tell the gonads to produce their hormones. So notice in this case, we have two levels of hormonal control. Hormone A controls the release of hormone B, and hormone B controls the release of hormone C. All of these are examples of hormonal control. So I want to erase that, and that, and that, and that, and that, and that. But I do want to release this. Hormonal control, I didn't leave that. Hormonal control is where one hormone controls the release of a second hormone. Oops. I need to get rid of that. Our second type of control is humoral control. With humoral control, it is the level of a chemical in the humors of the body, primarily in the blood, that control the release of a hormone. We've already talked about the classic example. The classic example of that is insulin. As our sugar levels spike from being at that party, we then release a massive amount of insulin as a result of that. But that's part of the problem, because as we've talked about, insulin is a very long-lasting hormone. It's a long-lasting chemical signal. So you have a spike of sugar, so you spike your insulin. And the insulin tells your body to put all this glucose into its cells. But it stays in the blood for a while, and the body puts more and more and more and more glucose into the cells until ultimately what happens?
Cells stop responding.
Okay, long term that can be an issue where the cell's not responding, but that's not what's happening to you that afternoon. What happens to you that afternoon after the party?
Blood sugar goes down.
Say again?
The blood and the blood sugar goes down.
Yeah, exactly. Instead of just bringing the blood sugar back to the levels where we normally would want it, because there's such a high level of insulin, the sugar levels continue to drop, right? And you become hypoglycemic and you get that sugar crash. Absolutely. Or you eat a very carb-rich food, spike your insulin, and then an hour and a half later, you feel hungry again. The reason you feel hungry again isn't because you are needing more nourishment. It's that a large amount of insulin has caused a drop in your blood glucose, and so you feel that as hunger as a result of that. Here, the example from the pretty picture is calcium levels. As we know, calcium makes cells do wonky things. So we need to keep a very, very tight control of calcium levels in the blood. So in this case, we monitor calcium levels. If calcium levels drop, our parathyroid glands release parathyroid hormones. which brings the blood glucose level, brings the calcium levels back up. And what was the third type of regulatory process again?
Neural.
Neural, absolutely. Here's a classic example. The medulla of our adrenal gland. What does the medulla of our adrenal gland release? Come on, I know you know. Adrenaline, absolutely, right? Bang, there's that big, huge, loud noise. Scares the bejesus out of you, right? But then you realize very quickly it's just the neighbor's car backfiring. However, while you instantly know that you're not under attack, right? Does your heart rate instantly go back to normal again?
No.
No, and the reason your heart rate stays elevated for a prolonged period of time is that our nervous system, in this case, our autonomic nervous system, or more specifically, our sympathetic nervous system, cause the release of that long-lasting chemical signal, that adrenaline, into our blood. So with neural control, there's some branch of the nervous system that controls the release of the hormone. So yes, great question. Absolutely on the exam as an essay question. Could I ask you the essay question to describe the 3 methods for regulating hormone levels in the blood? Absolutely. And would you need to, and would I probably ask for examples? More than likely. So would you need to describe these as we go through this? Yes. However, as you've seen from the handout, and we will go through this, for every single hormone, like for instance, if we're talking about adrenaline, for every single hormone, you're going to have to say, how is that hormone regulated? And if on the exam, I showed you this picture of the adrenal gland, and I had a big arrow that pointed right to this location. That's not a very big arrow, but you get the idea. I have a big arrow that points to this location, and I asked you, identify how hormone release is regulated from this gland. Be specific. What would your answer be?
Neural control.
And you would get partial credit for that on the exam. Remember also, when you see those words, be specific, that usually means more detail. You're right, it is neural, but I want the precise mechanism. What part, what branch of the nervous system? So neural control via the
Sympathetic nervous system.
Sympathetic nervous system. If instead, let's go back a picture. Here, if instead my arrow was pointing here, and I asked you to identify how the hormone is regulated in this gland, what would your answer on the exam to that be?
Humoral. Hormonal.
Hormonal via?
Thyroid. T3, T4.
Nope.
No.
Hormonally, humorally via? How is hormone release regulated in this gland? There you go. Humorally via calcium levels. Excellent. And let's go back one more pretty picture. Oops, that's not more. What if instead I said there? Identify how this gland is, release of hormone from this gland is regulated. And what would your answer to that be? Hormonally would be correct, but it's not via the thyroid.
Carrier pituitary?
There you go. Liz has got it. But again, no abbreviations on this exam, so you would need to spell it out. You would say humorally via thyroid stimulating hormone. All right? So again, I want the method of regulation, but I also want specifically what it is. And those are regulations. So again, absolutely, this is both lab and lecture type material. All right, questions on that?
So when you ask regulated by what, you mean regulated by which hormone?
If it is hormonally controlled, then yes, you need to tell me what hormone. If it is humorally controlled, you need to tell me what chemical in the blood. If it is neurally controlled, you have to tell me what branch of the nervous system. And like I said, don't worry, we will go through that handout together. So we will figure out the right answers as we go through it. So you'll know exactly what to say for each of them. But yes, the short answer is, however it's controlled, and I think I have to go back now to get those back. If it's neural, you need to tell me the branch of the nervous system. If it's humoral, you have to tell me what chemical. If it's hormonal, you have to tell me what hormone.
Okay.
I'm glad to hear that, Ashley. All right. But we're not quite to that handout yet. There's still a little bit more material we need to talk about, because not all hormones are built the same. There are different classes of hormones, basically based on what they are made up of. I keep talking about proteins because most of them are protein-based, but not all of them. So we will need to talk a little bit about that. However, it looks like this is a good point for our first break. It is 1:15, so let's take a 15-minute break. That means coming back at 1:30. I will start the recording at that time. All right, any questions before we take our break? All righty, see you in 15 minutes. All right. Any questions before we get started? All right, excellent. So as I mentioned, we need to talk about the classes of hormones. There's two major classes of hormones, and those are those that are amino acid based and those that are lipid based. Now, there are two major classes of hormones, and not surprisingly, there are two major differences between these two major classes. First is where they bind to the receptor. Amino acid-based hormones primarily bind outside of the cell. That is huge, so let's take that down. Outside of the cell, whereas lipid-based primarily bind inside the cell. Are there exceptions? Yes, but this is primarily, and so the sky is blue, and so that's the general rule of those. And then also how they modify the behavior of the cell, right? And by that, two overall basic effects, as we talked about, is to influence gene expression which is primarily what our lipid-based hormones do. And then the other overall effect, as we talked about, is to modify the metabolism. Increase it or decrease it. So those are the differences that we are going to see. And let's talk a little bit about each of the main cases, main types. Starting first with amino acid base, they are the more common ones, those that are amino acid based. And the primary difference between them is their overall size. Some of the smallest amino acid based hormones are what we call amino acid derived. They're actually formed by modified amino acids. The most common amino acids that are used modified this way are tyrosine and tryptophan. And here we see an example of it, one of our thyroid hormones, triiodothyronine, which of course with a name like that, you can be guaranteed you're gonna have to spell it at some point or another. The good thing is once you spelled it once, you can then abbreviate it T3. Of course, it's called T3 because it has 3 iodines bound to it. T4 is thyroxine, and it has 4 iodines bound to it, which is why they are commonly referred to as T3 and T4, but not by you. You will use their true names. Epinephrine, norepinephrine, melatonin, and like I said, there are many examples of the simple, basic, modified amino acid derived hormones. Some are a little more complicated. Our friend oxytocin is an example of what we call a peptide hormone. Peptide hormone is made of multiple amino acids bound together, typically somewhere between tens and thousands of amino acids put together. One of the interesting things about these are often made in an inactive state so that they don't affect the cell that's making them. And then once released, they become activated. Some examples of these are thyroid stimulating hormone, luteinizing hormone, antidiuretic hormone, human growth hormone, and there are lots of other examples as well. But some hormones are lipid-based. The most common type of lipid-based hormone is what we call a steroid hormone. And steroid hormones are made based on a modified cholesterol molecule. We learned about cholesterol, and here we see a picture of it when we were talking about our phospholipid bilayer. Remember, we talked about how it had a fluid mosaic in 430. And one of the things that helps to stabilize the movement of those phospholipids, helping to form the plasma membrane into an effective barrier, is by having these cholesterols embedded within the tail region of our phospholipid bilayer. And we were able to recognize those cholesterols because the cholesterol has these 4 carbon rings. So we use this 4 carbon ring as our starting point, and it becomes modified to produce hormones. We just put different side chains on it. Here we see an example of this. Notice we have 2 hormones here. Both of these are cholesterol-based, have those same 4 carbon rings to them. And as you can tell, the functional groups are only slightly different. That must mean that these hormones are very similar to each other. Yes, because they're so similar in their structure. They must be similar in their function as well. Does that make sense? Microphone is on, you can hear me, right? Do you believe that these 2 hormones are similar in their function or different in their function?
Sooner different.
You think they're different?
Different.
Again, different. All right, obviously. I'm asking the question. If they were similar, then I wouldn't be making the point of emphasizing it. Excellent. You guys get how the game is played. Absolutely. These are incredibly different. One of them is an estrogen, right? That's what turns girl people into women people. And the other is an example of a testosterone, which turns boy people into men people, right? These are obviously very, very different, and you wouldn't want to mix them up inside of the body. So very different while they may be very similar in their structure, those small differences are enough to have major impacts on the body. Now, on the exam, when we talk about, and notice if you looked at your list of hormones, is there a column to say whether they are steroid hormones or whether they're amino acid derived or whether they're polypeptides or anything along those lines?
No.
No, we're not going to have those that way because I don't need you to necessarily know that, but you do need to know which glands release which type of hormones. Steroid hormones are less common than the amino acid derived, and they are primarily produced by the reproductive organs, the cortex of the adrenal gland, and the kidney. So these are the only 6 organs that produce our hormone, steroid-based hormones, either the testes or the ovaries, the 2 adrenal glands, and just the cortex of them, and then the 2 kidneys. So every other gland that we're going to talk about produces amino acid-based hormones. And just like we talked about how adipose needs some type of water-soluble protein to be transported through the blood, so do our steroid-based hormones. They need that as well.
What about thyroid?
I'm sorry?
What about thyroid hormones? They're also from steroid.
Which hormones?
Thyroid. Thyroid.
Thyroid hormones? Thyroid hormones are amino acid based. Now, I know why you think that is because thyroid hormones do enter into the cell and they affect the cell inside of the cell. So it makes them think that they are lipid based, but they're actually not. Remember I said primarily, so they are special, but the thyroid hormones are indeed amino acid based.
Okay.
Yep. Alrighty. Now, those are our circulating hormones. But remember, we also talked about how there can be local hormones as well. And some of those local hormones are going to be lipid-based. An example of those are echocyanoids. Notice this echocyanoid only has a single carbon ring and a couple fatty acid tails. You could see why this would not be as stable for circulating throughout large parts of the body. So these are primarily used as local paracrine and autocrine hormones because they're not stable enough to be circulated throughout the entire body. Instead, they're going to have more local effects. And some examples of these, like I said, are our leukotrienes or some of our cytokines and things like that that we'll talk about. And then you guys will get those and talk about them more in 440 as well in micro. All righty. We talked about the different classes of hormones. And we also mentioned how there are different ways that they are going to affect the cells. So there are really changing the metabolism and gene expression are the three ways, right? So either going to increase or decrease enzyme activity, increase or decrease metabolism. And this, as I mentioned, is how our amino acid-based hormones are going to affect the cell. Now, they're able to do this thanks to a transmembrane protein. Because there is a transmembrane protein, to be able to influence some effect inside of the cell, right? These are amino acid based, so they're not lipid soluble. So, oops. So it's not easy for them to get inside of the cell. So instead, they use that transmembrane protein so that they bind outside the cell, but they affect some change inside the cell. Now, they can't do it by themselves. They require what we call a second messenger. My favorite example of this is to use the example, like right now, I'm in my office with the door closed, so I'm not bothering my family. So if the Amazon man who makes at least 4 or 5 deliveries at my house every day, just like I'm sure he does every day at your house, when he comes up and knocks on the door, if I'm back in the back of the house in my office with the door closed, talking loudly, am I going to hear him knock?
No.
No. But am I going to know when he shows up? Well, let me rephrase this. I happen to have 2 dogs. Am I going to know when there's a knock on the door?
Yes.
Yes.
Absolutely. I am going to know there's a knock on the door because while there's somebody outside who isn't able to come in, when they knock on the door, my dogs are going to go crazy and they're going to bark and they're going to jump around and they're going to knock the lamp off the table and make all this ruckus on the inside of the house that is going to make me aware of the fact that something happened outside. And that's exactly what a second messenger is. That second messenger is some signal inside the cell that can affect changes inside the cell based on something that happened outside. So we're going to need some type of second messenger since the hormone can't get inside. Now, the activation of this second messenger typically needs help, and that help comes in the form of G proteins. So here we see this nice illustration. Notice what's happening here. A hormone is binding to a transmembrane protein, a protein that is going through both levels of our phospholipid bilayer. It binds to the outside and it stays on the outside. But when it binds on the outside, it causes a change. It affects a change on the inside. There is this movable protein that is on the surface. This happens to be a peripheral membrane protein. Remember, peripheral membrane proteins are not attached to the plasma membrane, but are actually just associated with it. And when the hormone binds to it, forming that hormone receptor complex, it activates the G protein, and this G protein is able to move around inside the cell. Now, the G protein is not the second messenger. It is still a plasma membrane protein. It's associated with it, can't leave it though. So it's not the second messenger, but it's going to lead to the formation or the increase or decrease of the amount of second messenger. Let's actually see the actual mechanisms by which this occurs, and I think it'll help to make more sense. Now, as I mentioned, that G protein isn't the second messenger, but let's talk about what can be a second messenger. There are three primary second messengers. They're all special molecules. The first one is cyclic AMP, which is a modified adenosine monophosphate. So cyclic AMP, you'll write out once, and then you can abbreviate it, CAMP. This is the most common. And I'm going to describe for you the two most common pathways. And for both of those common pathways, cyclic AMP is the second messenger we will use. So the two pathways, that definitely will be potential essay questions that will be in the bucket your questions will be drawn from on the exam. Both will use cyclic AMP as its second messenger. But it isn't the only thing. Another energy molecule, cyclic GMP, a modified guanosine monophosphate, is a molecule that, like I said, you'll talk about more in micro, but it's also a energy molecule. Can also sometimes be a second messenger. And being a second messenger seems like a kind of wonky thing. So not surprisingly, sometimes calcium can actually be a second messenger as well. So these are the three most common second messengers. But like I said, for both the pathways you will be responsible for, the second messenger will be cyclic AMP. Well, like I said, cyclic GMP isn't going to be one of the pathways that you're going to be responsible for, so you don't need to use that term for the exam. Okay, so just cyclic AMP is fine. All righty. I will let you get away with AMP on this. So as long as you tell out cyclic AMP, I will let you get away with this. All right, here's the pretty picture for this, but let me go ahead. Let's do this on the whiteboard. So let's go to the whiteboard and talk about what's going to happen so we can draw this in a little bit more detail. Clear that. Move this up here a little bit. I can go down there. Sorry, I got to jut all of my windows. There we go. That works. All right. I'm just going to draw my, no, I guess I can't do, well, yeah, well, let's do it this way. I will draw our phospholipid bilayer as two lines. Appreciate that that represents that they are two layers of phospholipids, because then that will allow us to draw our transmembrane protein that goes through both of them. And this is our hormone receptor. And our hormone receptor, of course, has its binding site. So let's cheat a little more. There we go. Has a binding site for our hormone on its outer surface. This is outside. And of course, this is inside the cell. Oops. And as we mentioned, this hormone receptor is a transmembrane protein. And if we want to really be specific, it's a transmembrane integral. There we go. Integral membrane protein. Because it goes through both layers. And it is associated with a G protein. That is a peripheral membrane protein. Alrighty, so what happens first is that our hormone comes in and our hormone binds to the receptor. And when it binds to the receptor, it forms that hormone receptor complex. Again, this is occurring outside of the cell, so we bind. Let's make this smaller. So hormone binds. And when it binds that receptor, again, it forms the hormone receptor complex. And when it forms that hormone receptor complex, the hormone binds on the outside of the cell, but on the in cell, it activates the G protein. So the binding of the hormone outside the cell activates the G protein on the inside of the cell. Our G protein is a peripheral membrane protein. It's not attached to the membrane. It's just associated with it. And so when activated, the G protein can migrate along the plasma membrane. And so what's going to happen is this G protein is going to start to move along the surface of the plasma membrane until our G protein makes contact with a second integral membrane protein. And not just any integral membrane protein, but a special membrane protein bound in just one level of the phospholipid bilayer called Adenylate Cyclase. Now, with a name like Adenylate Cyclase, what do you think Adenylate Cyclase is?
An enzyme?
An enzyme. It ends with the suffix ase, so this is an enzyme. This is an enzyme. When our G protein makes contact with it, then what happens is this enzyme is activated. And when the G protein is activated, it's an enzyme, it facilitates a chemical reaction. And what it does is it converts ATP, our energy molecule, into cyclic AMP. So basically what happens is we have some ATP, and cyclic adenylate cyclase converts this ATP. Oops, ooh, I like that one. Let's do that one. Into cyclic AMP. And cyclic AMP, remember, is our second messenger. So notice this enzyme makes more of our second messenger. Or you can think of it as the concentration of the second messenger increases. All right. Now,
so the G protein activates the enzyme?
Activates the enzyme, exactly. The G protein, when it connects to it, it activates the enzyme, and suddenly we have a lot of cyclic AMP. And here's the other key to this process. Notice how many hormones had to bind.
One.
Activating how many G proteins? One. Activating how many Adenylate Cyclases? One. And that one enzyme, is it going to just convert one ATP into one cyclic AMP?
No.
No. Notice one of the keys to this process is this process it's a cascade effect. Right? Notice it goes step by step by step by step. That's not a new concept to us. We've talked about cascade effects before. But in this case, it is a cascade effect with amplification. One hormone binding to one receptor can produce hundreds, if not thousands, of new cyclic AMPs. So just one hormone binding to one receptor has the potential to give us a massive effect. So we get this massive amplification of this process, giving us lots and lots of these cyclic AMPs. Of course, the question becomes, what is it that the cyclic AMP does? And primarily, what this cyclic AMP does is our cyclic AMP activates a special protein, a special enzyme inside the cell. And this special protein is called a kinase. Let's say it this way. Special proteins, plural, called kinases. And then these kinases then go around and basically can do a number of things. They can turn other enzymes on. They can, whoops, hold on, move that here. Kinases can change the membrane permeability. By opening or closing channels. And overall, it increases the metabolism of the cell. And so the overall effect in this pathway is to excite the cell or increase metabolism. And this is our first possible pathway, an excitatory So pathway 1 is an excitatory pathway where it increases the metabolism of the target cell in a cascade effect with amplification. Now, I've done an amazing job of drawing this picture, but let's look at the pretty picture from your textbook. Notice we see the exact same thing going on here. Our hormone binds to the protein receptor, which is a transmembrane protein, which activates the G protein. Now notice our pathway here is a little wonky, but again, this is a simplified illustration. The one from your textbook is really nice. We'll look at the picture from your textbook in just a minute. G protein is activated by the hormone. G protein activates the adenylate cyclase, which converts ATP into our second messenger, cyclic AMP. Cyclic AMP activates our kinases, which activates our enzymes and opens our ion channels, and the metabolism increases. Yes, great question. In this pathway, this receptor to this hormone, this G protein always attaches to adenylate cyclase. However, different hormone, different receptor, different G protein, is it going to bind to a different enzyme? Absolutely. So again, here we have the excitatory pathway. This is our most common excitatory pathway. It increases the metabolism. Hormone binds the receptor outside of the cell. And again, we can't emphasize this enough. This occurs outside, whereas the G protein is activated inside. You don't mention that on your essay question, you will lose points for that. The big key to these amino acid hormones is they cannot enter the cell. That's why they need the second messenger. So it binds outside, activates the G protein inside. G protein activates adenylate cyclase, converts ATP into cyclic AMP, our second messenger, which activates kinases, which is going to phosphorylate proteins. Like we said, it's going to open ion channels. It's going to activate enzymes. We are going to get an increase in the metabolism of the cell and a cascade effect with amplification. Questions on that pathway? All right, excellent. So then, oh, and as promised, here is a better look at the picture from your textbook. Again, notice, and even your textbook gets it a little bit off. This adenylate cyclase is really, it is an integral membrane protein, but it typically doesn't go all the way through both phospholipid bilayers. But here we see more of that migration we were talking about. Hormone binds outside, activates the G protein inside. G protein migrates along the phospholipid bilayer till it finds the adenylate cyclase, activating this enzyme, converting ATP into cyclic AMP, activating the kinases, and kinases activate our enzymes, open up ion channels, can stimulate cellular secretion. The overall metabolism increases. All right.
Do we have to know what GDP is and GTP?
No, you don't have to know how it's activated. You just need to know that it activates it. Okay. Questions on this first pathway? All right, then let's look at our second pathway. And let's go to our whiteboard for this again. All righty, it's the easiest way for me to get rid of all of this. I guess it might as well just clear all of it. There we go, phospholipid bilayer, hormone receptor, G protein, integral membrane protein. Excellent. All right, let's do this again. This time we are going to focus on the primary inhibitory pathway. And of course, the job of the primary inhibitory pathway will be to lower the metabolism. All right. It starts the same way. Now a different hormone, hormone B, binds to a different receptor, forming that complex. When it forms that complex is going to activate a G protein. Hopefully I don't have to write all the definitions of things like we did last time. When the G protein is activated on the inside of the cell, again, let's do this inside, emphasize this, binds outside. And that says outside. Activates the G protein on the inside, and that G protein migrates along the plasma membrane until it again comes in contact with an enzyme. However, this time it is a different hormone, a different receptor, a different G protein, so it activates a different enzyme. And the enzyme that is activated here is phosphodiesterase. Phosphodiesterase. Again, still an ACE, it's still an enzyme. However, this time when active, and the good news is once you write out phosphodiesterase once, you can abbreviate it PDE. When active, PDE converts cyclic AMP into just the simple energy molecule AMP. So what happens is that our cyclic AMP is converted into just AMP. And so when this enzyme is active, what happens to the levels? No, phosphodiesterase is an enzyme, not a receptor. The receptor, this over here is the receptor. Both are integral membrane proteins, but the receptor is what the hormone binds to. Phosphodiesterase is an enzyme that converts cyclic AMP into AMP. Now, what did we say our second messenger was? What's our second messenger? Cyclic AMP. Cyclic AMP is still the second messenger. Oops. It's still the second messenger. However, when phosphodiesterase is active, what happens to the amount of cyclic AMP we have inside the cell?
Decreases.
Exactly. So when the phosphodiesterase is active, the levels of cyclic AMP decrease, right? Or there's a decrease in concentration of cyclic AMP. And if there is less cyclic AMP, what happens to the number of kinases we can activate?
Decreases.
Activates fewer.
Yeah, fewer kinases are activated. And if fewer kinases are activated, let's just do it this way. Put that there, like that. And if fewer kinases are activated, then what is going to happen to the number of enzymes that can be turned on?
Lowers.
Decreases.
Yeah, fewer enzymes are activated. Fewer channels are open. And overall, we get a decrease in the number of chemical reactions. We get a decrease in metabolism. as a result of this. And this is our primary inhibitory pathway. Questions on this?
So the other one's called the primary excitatory pathway.
Yep, and this is the primary inhibitory pathway. So again, still have the same starting point. Hormone binds outside of the cell, activates a G protein inside of the cell. But this hormone and this G protein activate phosphodiesterase. And phosphodiesterase breaks down cyclic AMP, converting cyclic AMP into just AMP. First of all, not a silly question at all, but absolutely. You are correct. When people talk about having a fast metabolism, they're typically not talking at the cellular level like this. No. Gdp and Gtp, remember, are just energy molecules. You don't have to worry about what those are. But those are just energy molecules that, again, they're lower energy molecules than ATP and ADP. So it's guanosine triphosphate and guanosine diphosphate. Like I said, you guys will talk about that in micro. Or if you ever take like a 400, you'll probably talk about that in there when you get to the cyto stuff, but we're not going to worry about that in this class. All right, and then obviously that means we have less of our second messenger, fewer kinases, decrease in enzymatic enzyme, and metabolism decreases. And once again, notice we are getting a cascade effect with amplification. Because again, one hormone binds to one receptor, activates one G protein, activates one phosphodiesterase, but that one phosphodiesterase can break down many cyclic AMP. And if it breaks down many cyclic AMPs, then we have a dramatic decrease in our metabolism. So notice both of these amino acid-based pathways both have this cascade effect with amplification. All right, so we have seen how our amino acid based, and again, that's the key, amino acid based pathways, two of them, one excitatory, one inhibitory. Questions on that? All right, so then the question is, what the heck is it that our lipid-based hormones do? Well, our lipid-based hormones are the ones that we talked about are responsible for gene expression. All right, and luckily, way back in 430, we had learned about what exactly gene expression is. Most genes carry the instructions for doing what?
Making proteins.
Making proteins. And how many steps are there in the protein synthesis process?
2.
Excellent.
Transcription.
There you go, absolutely. 2 steps. Transcription and translation, in what order do they occur in?
First transcription, then translation.
Transcription and then translation. Where did transcription occur again?
In the nucleus.
Excellent. Where did translation occur again?
In the rough endoplasmic reticulum.
Excellent. And more specifically at a ribosome. But you are absolutely correct. Most ribosomes are located in that rough endoplasmic reticulum. And we learned about start codons and stop codons, and we learned about all of that type of stuff. But let's go back to transcription again. Anyone remember how many steps there were in transcription? 3, absolutely. Do you remember what they were? Anyone remember the 3 steps of transcription?
Initiation.
Okay. And then elongation, and then termination.
Termination.
Right? That sound vaguely familiar? Yes, no, maybe. And remember, in fact, initiation, right? We needed to find a special sequence in the DNA. Anybody remember what that special sequence of the DNA was called? There we go, excellent. A promoter region. Excellent, we have that initiation or promoter region, right? So somewhere on the DNA, here we have this strand of DNA. And on this strand of DNA happens to be the particular sequence that gives me the instructions for making a protein. And at the very beginning of that, we had a special promoter region. And at the very end of that, we had, oh, hold on, let's change colors. A termination sequence, so that our RNA polymerase would know where to bind on the DNA, where to uncoil it, and where to start reading. Now, if you remember when we talked about protein synthesis in 430, we have 2 problems. The first problem we have is, does anybody remember what percentage of our DNA actually codes for proteins? There we go. Only 1.5% of the DNA codes for proteins. So if you think about it, if I were to throw a dart at my bowl of spaghetti that is DNA, what are the chances that I'm going to hit the gene I want? Heck, what are the chances of me hitting a gene, let alone the gene I want, let alone the promoter region of the gene I want? Absolutely slim to none. Absolutely slim to none. Very, very unlikely that it's going to happen. And the other issue is that my toenail cells, my eyeball cells, my liver cells, do they have the same DNA or do they have different DNA?
Same.
Same. So if they all have the same DNA, how do I tell my toenail cells to make toenail proteins and my eyeball cells to make eyeball proteins and my liver cell to make liver proteins? How can I tell them to make different proteins and when to make those proteins and even find those if I've got this shot in the dark of being able to find it? Well, if you remember way back in 430, the way we said this was with hormones. Hormones are what tell the cells what protein to make and how. And this is how it actually occurs. I'm not going to bother drawing this on the other screen. We'll just use it here so we can see what happens. I'll highlight it as we work our way through. Notice, because lipid-soluble hormones are lipid-based, or let me say that the other way. Lipid-based hormones are lipid-soluble. Is a lipid-based hormone going to have any problem getting into a cell, getting through that plasma membrane?
No.
No, it's not going to have any problem at all. So basically, it goes straight through the plasma membrane just by simple diffusion. Once in the cell, and this can actually occur in the cytoplasm, Or in some cases, it can actually occur in the nucleus. This is getting too big. I need to make it a little smaller. Either in the cytoplasm or in the nucleus, our hormone binds to the receptor. And again, when our hormone binds to the receptor, it forms that hormone receptor complex. So we have some hormone receptor. And our hormone comes in and binds to it, forming that hormone receptor complex. Once this complex is formed, it either enters into the nuclear envelope, or if it already occurred inside the nuclear envelope, it just stays in here. And what this hormone receptor complex does is it actually binds to the DNA at the start of the promoter region. Oops. So it actually comes and binds to the DNA for the gene we want to express right at the beginning. And basically what it does is it forms an anchor or an attachment point. And this attachment point is what allows the RNA polymerase to find the promoter region and to start transcription. So what's going to happen is our big, huge RNA polymerase is going to find this promoter region now thanks to that complex, and it is going to be able to read the DNA, make our messenger RNA, and once that messenger RNA is made, that messenger RNA leaves the nucleus going to the rough endoplasmic reticulum, where it is translated into a sequence of amino acids, where it then goes to the Golgi apparatus to be cut, folded, modified, and made into a protein. This hormone receptor complex allows us to solve our two problems. By binding to a specific place on the DNA, it tells the RNA polymerase which gene to read and when to do it. Because it can't bind to it if that complex isn't there. So only when the complex is there can my RNA polymerase bind, make the messenger RNA necessary, and start our protein synthesis. So let's think about this again. I'm going to erase all of this because I've kind of made a mess, but I want to kind of emphasize a couple things. So again, we form the complex. Inside the cell, or actually in some cases, inside the nucleus. Right. Binds to the DNA at the right gene, and it triggers initiation of the RNA polymerase. And then that whole process we talked about in protein synthesis occurs, right? We make our messenger RNA. Messenger RNA goes to the rough endoplasmic reticulum for translation. After translation, we have a sequence of amino acids. And that goes to the Golgi apparatus or the Golgi complex, where it is cut, folded, modified, added to other subunits, etc., to make a functional protein. Then once it makes that functional protein, remember the Golgi apparatus, or the Golgi complex, packs it in a vesicle. And if you think about it, it either is released via exocytosis, or it gets expressed on the plasma membrane. It becomes a part of the plasma membrane. If these concepts are foreign or confusing to you, I strongly encourage you to go back to chapter 3 when we talked about the cell. And once we do that, go back and read those to understand what happens with the Golgi apparatus, what happens with these hormones, what happens with these proteins as they're either released via exocytosis or they're expressed on the surface. Now, compared to amino acids, where we're basically turning enzymes on and off, is this a faster process or a slower process?
Faster.
Faster? Translation, transcription, modified by the Golgi apparatus, packaging in a vesicle, moving to where we want. Is that faster than turning switches on and off?
Slower.
Yeah, it's a much, much slower. This is a relatively slower process than the amino acid-based ones. But think of it this way. The effects of this can be much more powerful. Yes, amino acid hormones can turn enzymes on and off, can open or close channels. But here, we could make a new channel. We could make some new channel that this cell has never had before and express it on its surface. Or a new receptor that allows it to respond to a hormone that it's never been able to respond to before. Right? Some of the changes that can be made by this process, while it's slower, some of these changes can permanently alter the cell. So while it is a relatively slower process, it is incredibly powerful. The ability to make proteins and express those proteins. Right, as we talked about it, our genes are what determine what we are. Whether you're right-handed or left-handed, whether you have blonde hair or brown hair, whether you are predisposed to heart disease or not predisposed to heart disease, right? Your blood type, everything. Everything about you is expressed in these genes. And so by expressing these genes, making these proteins can have a huge, powerful, long-lasting, long-term impact on the function and the structure of this cell. Questions on that? All right, so again, hormone, because it's lipid soluble, because it's lipid based, passes through the phospholipid bilayer by simple diffusion, binds and forms that hormone receptor complex. Complex binds to the DNA, and that gene expression occurs. And again, here's the simple picture. Make a new messenger RNA, make a new protein. Protein can alter the cell's activity. This is that simple illustration. I believe this is the one from your textbook. I like it because the hormone receptor complex reminds me of candy corn, and it's almost Halloween, so I like that. Binds, messenger RNA, ribosome, makes our sequence of amino acids. That then has to go. Don't forget about needing to go to the Golgi apparatus, right? Because if you don't go to the Golgi complex, you don't get the fully functional protein.
And you want us to mention it?
Absolutely. Right? Because that's the importance. Here we are expressing this gene, and we express this gene by making functional proteins. So you can't forget about the step of transcription, translation, and modification in the Golgi. Now, do you have to describe every single step of translation, starting with those start codons and making a bond and breaking a bond and stop codons and methionine and all of that stuff that goes along with it? Do you have to do all of that? No, right? You had to learn that in 430. But do you need to get all the way to the point where you have a functional protein, and that functional protein is either going to be expressed on the surface or released via exocytosis? Yes. So take it all the way to the point where you have a fully functional protein doing its job. Excellent. And like I said, do you think all three of these essay questions are in the bucket of questions you could possibly have on the exam? I sure do. Yes, I definitely think you should, or at least call and yell or email and yell at your 430 instructor. Absolutely. All right. Excellent. So, perfect timing. We're at 2:30 right now. We have gone through the mechanism. So now what we're going to start doing is looking through the list of those endocrine hormones, looking at the list of the organs, looking at the histology of these things. And again, like I talked about, so if you haven't printed out the list yet, I strongly encourage you to do that now. First and foremost, shame on you if you haven't. But let's take a look at those and start going through them. We're going to talk about the glands, what hormones are produced that you're responsible for. So many of these glands produce more than what we're listing. On the list are the only hormones you're responsible for on those glands. Know the classes versus specific hormones when appropriate. Know their targets, know their effects, and know how they're regulated. And so when we come back, this is where we will start. We will start with this mechanism, working our way through that table, making sure we understand it. All right? So, like I said, it is 2:30 now, so we'll take a 15-minute break, come back at 2:45, and at 2:45, we will restart. Excellent. All righty. All righty. Any questions before we get started? So, if you've looked at the list at all, not surprising, one of the primary glands of the endocrine system is our pituitary gland, or what is also known as the hypophysis. This gland is known as the master endocrine gland because it produces over a dozen different hormones, and many of them are things like thyroid stimulating hormone. And what did we decide thyroid stimulating hormone did? These are the easy questions.
Like the thyroid?
Yeah, it stimulates the thyroid gland, exactly. So many of the hormones that it produces actually regulates and controls other endocrine glands. So it is definitely the master endocrine gland, and for a very long time, it was thought to be the command center of the endocrine system. Now, of course, with a term like that, thought to be the command center, obviously we now know that it isn't, but it's only 20 years ago or so that we actually found that to be the case. So if you think about in how long we've been studying the body for, to only know for the past 20 or so years that there's this major change in how this works is something that is pretty darn impressive. So this is our master endocrine gland, and it primarily has two components to it. When we look at it histologically, we'll see this, but all the pretty pictures show this nicely as well. The first part is what is known as the posterior pituitary, or the neurohypophysis. As the name would indicate, this posterior pituitary is comprised of nervous tissue. Now, There is one minor problem with that. If you think about it, nervous tissue really isn't glandular tissue. So what's special about the posterior pituitary is the one gland that we're going to talk about that actually does not make its own hormones. Instead, it just releases hormones, and those hormones are made someplace else. I think maybe there's enough room for that. Let's see. Oops, wrong direction. Bingo. Okay, so here, let's do this. There we go. So made of nervous tissue, does not make its own hormones. Instead, those hormones are made in the hypothalamus, and it is connected to the hypothalamus by the infundibulum. If we were in the classroom, we'd be able to look at the brain models, and when you look at the brain models, you see there's this pink M&M connected to the bottom of the brain models. That is our pituitary gland, and it connects to the infundibulum. And we have some pretty pictures that we will use to look at these in just a minute. The second part is the Adeno Hypothesis. The Adeno Hypothesis is what is also known as the anterior pituitary. And this one is glandular tissue. So being glandular tissue, it both makes and releases its hormones. And that's in fact what most of our endocrine glands do. As I mentioned, the posterior pituitary, the neurohypophysis, is the only gland we're going to talk about that doesn't actually make its own hormones. So everything else that we're going to talk about both makes and releases its hormones. And it is glandular tissue. Now, the adenohypophysis is much larger than the neurohypophysis. The anterior pituitary is much, much larger, and it is divided into three major parts. So what we can do here is look at a little bit of histology. Now, before we do this, let's actually cheat for a second. I should have set this up during the break. I wasn't thinking. Let's do this real quickly and see if we get lucky. What I want is... That's not what I want. That's what I want. Perfect. All right, these will show you. That's one of the things I want. Yeah. And then the other one I want. I'm being nervous, isn't it? Perfect. All right, these are what I want. So here is our half-head chart. We have seen this before in this class and in the classroom itself. And as we look at this, one of the things that you will notice, and let's use the highlighter so I don't block anything, and use red to make it stand out. This structure right in here is the, so that doesn't show up nice at all. Pink, nice and bright. There we go. That little nugget hanging out right down in there is the pituitary gland. We talked about that in 430 because as we saw, there was this nice U-shaped indentation in the bone. Anybody remember what that bone feature was called?
Sella turcica.
Sella turcica, excellent. Did we have another name for it? Sella turcica is Turkish saddle because it has this really high front end back to it, but excellent. The other name, remember, was the hypophyseal fossa. A fossa is an indentation, and this indentation is for the hypophysis, the pituitary gland. So the hypophyseal fossa. So notice that indentation. And anybody remember what bone that was, that is a bone feature of?
Sphenoid.
Sphenoid, excellent. Notice when you look closely at the illustration, though, you can see that they've done a nice job of drawing this dividing line and having a dark part and a light part to it, representing the anterior and the posterior pituitary. And that is, of course, I'm sure as you remember from 430, this region up here is the hypothesis, the hypothalamus, and the hypothalamus connects to the hypophysis by the pituitary gland. The classroom also has this nice illustration, a chart here that shows this as well. So notice here we get a nice view of the infundibulum, this stalk-like structure that connects the posterior pituitary or neurohypophysis to the hypothalamus. And then this larger portion to the front is the anterior pituitary. So if you see, here, with the histology, we've actually taken a slice through that structure. We can see that anatomy here as well. Again, this up here, no, I wanted that. There we go. This whitish stalk-like structure is the infundibulum. Notice this tissue here is lighter in color, more whiter in color. That is the nervous tissue. Why might nervous tissue look lighter in color?
No blood cells?
No, we need blood, but myelin, excellent. Right, and since this is in the central nervous system, which neural glial cell would be providing that myelin sheath? Well, you know it can't be the Schwann cells, because if it was the Schwann cells, I wouldn't be asking the question, because Schwann cells is way too easy to say. There we go. Instead, it is the oligodendrocytes, absolutely. Oligodendrocytes. So nervous tissue is typically lighter in color because of that myelination to it. So here is this nervous tissue, that posterior pituitary, and then we have the much, much larger anterior pituitary to the front, which is comprised of glandular tissue. Now, this is much larger, and it is divided into 3 distinct regions. The portion of the anterior pituitary that wraps around the infundibulum is known as the pars tuberalis. The largest portion, the portion that is most to the front, is the pars distalis. But if you notice closely, even in this very low magnification view, you see that this transition region right here, especially kind of like in this area, looks kind of different from the rest of the anterior pituitary. And this region is what is known as the pars intermedia. The other place that they do a nice job of showing this is on that chart. So if we go back to that picture of the chart, hopefully they have this here. We go there. Ah, perfect. That was what I was looking for. Notice what they've done here is taking a very high magnification sagittal section of the hypophysis. And what you see very clearly over here is the nervous tissue. It's more lighter in color. And we have these kind of star-shaped, asteroid-shaped looking cells. These are the neuroglial cells that are going to play a role in helping to store and release the hormones from the posterior pituitary. And anybody know what the name of these cells found in this region, the nervous region, might be? If only I had a handout that had the anatomy and histology I was responsible for. It wouldn't be astrocytes. Ah, there you go. Pituicytes, absolutely, the pituicytes. These cells found in the nervous region are the pituicytes. Notice here and here, we have more glandular looking structures. And these glandular looking structures collectively, so this is the dividing point right here, everything back here is the anterior pituitary. But notice it's not all the same. This transition region from the posterior pituitary to the anterior pituitary, that pars intermedia, is a very unique region of the pituitary gland where only one hormone can potentially be produced. So this right here is kind of, it is part of the anterior pituitary, but it is a special part of the anterior pituitary where only one hormone and one hormone only can be produced. And looking at your list of hormones, which for the anterior pituitary, which one do you think it might be? Is there something different about one of the hormones listed from the anterior pituitary on your handout that I encourage you to have printed out for this part of the lecture?
Melanocyte stimulating hormone.
Yeah, what's special about the melanocyte stimulating hormone? What made you say that one? What's different about it on the list? It has an asterisk next to it. Yeah, it has an asterisk, exactly. And that's why it has the asterisk, because that one can only potentially be produced here in the pars intermedia. All right, so notice you want to make a note on that on your handout that it is only potentially produced in the pars intermedia. What do you think the target of a melanocyte stimulating hormone is?
Skin.
Okay, but could we be more specific?
Melanocytes in the skin.
Yeah, how about melanocytes in the skin? Absolutely, that would definitely be the target. And what do you think it would stimulate your melanocytes to do?
Produce color.
Melanin. There you go, excellent. What does melanin do?
Produce color in your skin and your hair.
Right, absolutely. So for instance, if I was sitting here in my chair and a bear with an axe walked into the room, I would be able to release melanocyte stimulating hormone, which would go to the melanocytes of my skin, causing me to change the color of my skin so that I blended in with my chair and I would be able to hide from that bear with an axe, right? So as long as I don't move, I would blend right in with that chair and the bear with the axe wouldn't notice me.
Unless you're a chameleon.
True, you're absolutely right. Are there things like frogs and other types of lizards and other things that are capable of doing that? Absolutely, and that is one of the ways that they do that. However, we don't work that way. So while there are cells in here that are that can potentially make melanocyte stimulating hormone, is that something we normally do on a day-to-day basis? No, absolutely not. In fact, in a normal healthy individual, we really don't make this hormone. Now, if you had a tumor in the pars intermedia of your pituitary gland or some type of other pituitary abnormality, might you start producing this and might your skin take on a darker pigmentation? That could be an indication that you've had some type of pituitary problem. Yes, absolutely. But this is not a hormone that we normally use and functions in our normal body. So clearly we know where it's produced, we know what its target is, we know what its function is, but do we really have to worry about regulating this one? How this one is regulated, is that a question I'm gonna ask on the exam? No, so you can put an X in that box because we're not gonna worry about how this one is regulated. Okay? Great question. No, albinism is not a problem with this pituitary gland. It has more to do with the genes. Remember back in 430, we said there are at least 4 genes involved in what color pigment your cells produce. Each one has a light and a dark version. There's at least 4, there may be more, but there is also a 5th gene as well. And that 5th gene is the one that turns the melanocytes on. If you have a mutation of this gene, it doesn't matter what color pigments you get from those other genes, your melanocytes don't produce any melanin. So albinism is actually a gene mutation where your melanocytes are not capable of producing melanin. So it doesn't have anything to do with the pituitary. Possibly some dark patches, although because it's a hormone that can get in your skin and go most places, while there may be some irregularities to the color, typically you would see a darkening of the skin everywhere. And that's kind of one of the indications. Even under your clothes, under your underwear, places that don't normally see a lot of sun, you would get dark pigmentation to the skin. It wouldn't necessarily be completely uniform, but you would see it everywhere throughout the body. And again, it's not a common condition, but that is that one. So that is the one special part of our hypothesis with that one unique hormone that, again, isn't normally influence and affecting things in our body, but when there's an abnormality, we can see it that way. Okay. So here we see it with the illustration. We even saw it at a lower magnification, how there's a little bit different something in that pars intermedia versus the other regions of the anterior pituitary. And here we see in a light microscopy. In a light microscopy, again, here we can see the pituicytes of our posterior pituitary. Here we can see the glandular tissue of our anterior pituitary. And here we have this thin, I'll box it, kind of wonky transition region here. That would be the pars intermedia. So all of these are examples of ways, both in illustrations, light microscopy, and models, we could see the three distinct regions of our anterior pituitary. Although again, like I said, this is the two regions, and then obviously the pars tuberalis would be superior to this. But this special region, the pars intermedia. All righty, we will talk about the hormones of the pituitary gland in just a minute. But like I said, it's the master endocrine gland, and for hundreds of years, we thought it was the command center of the endocrine system. And then, just over 20 years ago, we realized, wait, we got it wrong. There is a power behind the figurehead. It's kind of like in politics, right? The president is the person that you see out in the front, right, making the decisions, commanding and leading us. But all of us know that really behind the scenes, the Illuminati controls everything. Right? There's that power behind the scenes. And that's what the hypothalamus is. The hypothalamus is the power behind the scenes, right? The hypothalamus is the Illuminati. It is a combo platter of both neural and endocrine tissues. It is a part of our central nervous system. And as we learned in 430, the hypothalamus is a major regulatory organ. It regulates our thirst and our hunger, our body temperature, much of our limbic system. It's this massive regulatory region that plays a tremendous role in regulating many of the basic biological processes in our body. But its other main function is to control the endocrine system. Now, to control the endocrine system, we do have one minor problem. If it is going to control the anterior pituitary and the posterior pituitary, right? If our hypothalamus controls the pituitary gland, the anterior pituitary and the posterior pituitary are made up of two different types of tissue. One's glandular and one's neural. And can we necessarily control glandular tissue and neural tissue the same way?
No.
No. So we're going to need two mechanisms for controlling the pituitary gland. Let's switch to our whiteboard and see how this might work. So we have our hypothalamus up here, and then down here we have our pituitary gland. This over here will be our anterior pituitary. Actually, let's not do that. And this is our posterior pituitary. And as we know, the posterior pituitary is connected to the hypothalamus by the infundibulum. So let's throw some quick labels on here. Infundibulum. Excellent. And actually, yeah, we'll put that right there, right next to it. That'll work. So we'll put that right there. This is our anterior pituitary. Or Adeno Hypothesis. Again, remember those terms are interchangeable. And this is the posterior pituitary. Okay. Now, what type of tissue is the posterior pituitary again?
Neural.
Nervous tissue, excellent. Neural tissue or nervous tissue. Excellent. And our anterior pituitary is, of course, glandular tissue. Remember, I told you one other special thing about our posterior pituitary. It does not make its own hormones. It just stores and releases them. So we have special cells and what were those special cells that we said inside here that are going to store and release the hormones? Pituicytes. So we have these pituicytes, and these pituicytes are going to store and release the hormones. So what happens is up here in our hypothalamus, we have neurons. Those neurons are going to make the hormones, basically the neurotransmitters. And then they are going to carry them down their axons to give them to the pituicytes, and then also tell the posterior pituitary when to release them. So basically up here, because this is the central nervous system, what would the structural classification of this neuron be? Excellent, thank you, Alexa. As a neuroscientist, the fact that nobody jumped to say that faster did hurt my heart a little bit. Excellent, we have that multipolar neuron with its singular axon that is going to come down, and it is going to release those hormones to the pituicytes, and the pituicytes store and release them. So when I want these pituicytes to release the hormones, I produce an action potential. That action potential spreads down, and I tell these pituicytes to release them. And of course, they're released into the blood. Right, into the veins, which carry it to the heart. And then from the heart, everywhere in the body. What kind of control would this be then for our posterior pituitary? What type of regulation of the hormones that are produced and released here, what would that be?
Neural.
Excellent. So notice for all of our posterior pituitary hormones, they are regulated via neural control from the hypothalamus. And just out of curiosity, how many posterior pituitary hormones are you responsible for?
2.
What are they?
Antidiuretic hormone and oxytocin.
There you go, oxytocin and antidiuretic hormone. So for both oxytocin and antidiuretic hormone, what can you write down in the box for how they're regulated?
Neural control.
And you'd get partial credit for that on the exam.
Yeah, hypothalamus.
There you go. Neural control from the hypothalamus or via the hypothalamus. Excellent. Excellent. Hopefully that's pretty straightforward. Questions on that? Now, notice this works for two reasons. The posterior pituitary is neural tissue, and the infundibulum is neural tissue. So those axons can attach directly to them. The problem is over here in the anterior pituitary, we have glandular tissue. And since we have glandular tissue, we need to figure out a different way to communicate. Oh, no. Let's go back to leaving that red. Okay. We need a different way to communicate. And luckily, the way we're going to communicate here is via the blood. Hypothalamus communicates with the anterior pituitary via a special blood vessel pathway. If I remember correctly, we talked about a special blood vessel pathway associated with our digestive system. Right? We had capillaries in our digestive system where exchange of materials took place. But instead of sending that blood straight back to the heart, we sent it to a second capillary located in some special organ. What was the special organ that received the blood first? The liver. And what did we call that special blood vessel that carried the blood to the liver?
Hepatic portal vein.
A portal vein, exactly, the hepatic portal vein. And that's exactly what happens here. Again, this is nervous tissue. So there are going to be some neurons that are going to produce and release their hormones. And they're going to release these hormones into a capillary bed. Just draw that like that. This is this capillary bed. This capillary bed is then going to carry this, instead of directly back to the heart, carries it to the anterior pituitary where there is a second capillary bed. And not surprisingly, this blood vessel is called pituitary portal vein. So up here at the first capillary, what happens is, and I'm going to change colors now just to emphasize this, our hypothalamus produces two classes of hormones. Classes of hormones mean that there are many types, almost a dozen different types. And these two primary classes are what are called releasing hormones and inhibiting hormones. What do you think releasing hormones do?
Release hormones?
Yeah, they tell the anterior pituitary to make and release more hormones. And guess what inhibiting hormones do?
Inhibit this process.
Yeah, tell the anterior pituitary to make and release less hormones. So those are the two main classes of hormones. And there are lots of specific types. For instance, there is a thyroid stimulating hormone, releasing hormone, and an inhibiting thyroid stimulating hormone. Guess what the releasing thyroid stimulating hormone tells the anterior pituitary to do?
And release it.
Make and release more thyroid stimulating hormone. And guess what the inhibiting thyroid stimulating hormone tells the anterior pituitary to do?
Inhibitory stimulating.
Yeah, make less and release less of that hormone. So basically, every hormone that the anterior pituitary releases, for every hormone that the anterior pituitary releases, the hypothalamus has two, a releasing hormone and an inhibiting hormone. So it releases, and let's go simple. It releases a releasing, thyroid stimulating hormone. Ugh. So this is released by here, and it enters into the capillary. It travels down the capillary to the anterior pituitary, where it comes out, releasing thyroid stimulating hormone, and tells the glandular cells, here's one of those glands. Inside of my anterior pituitary. And it tells this gland to make thyroid stimulating hormone. That thyroid stimulating hormone then enters into the capillary, and then this capillary carries the blood back to the heart and is spread throughout the body. There you go. So notice every single hormone in the anterior pituitary, with the exception of a melanocyte stimulating hormone. Now, is the melanocyte stimulating hormone possibly regulated hormonally? Maybe, probably, because everything else in the anterior pituitary is, but we don't know that for a fact and we don't care. But so every other hormone, you're right, on the list is regulated hormonally. But don't just say from the hypothalamus, you need to say regulated hormonally by releasing and inhibiting hormones from the hypothalamus. So let's write that out. All anterior pituitary hormones are regulated hormonally via releasing, inhibiting hormones of the hypothalamus. There you go. It's a tiny bit of a mouthful to say, but learn it once and use it, what, 7 times, 8 times, however many anterior pituitary hormones you're responsible for. Great question. Absolutely. So we can go a step backwards. Our hypothalamus is primarily nervous tissue, part of our central nervous system. It releases 2 major classes of hormones, and those are the releasing hormones and the inhibiting hormones. What are their targets?
Anterior pituitary gland.
Anterior pituitary, excellent. Releasing hormones increase the hormones made and released. Inhibiting hormones decrease the number of hormones made and released. So then absolutely the question becomes, how is the hypothalamus regulated? And as a central command center of the central nervous system, it is going to be regulated how? How do you think it's going to be regulated? Primarily neurally, absolutely. It is going to be regulated neurally, and I would say by the central nervous system. However, the other thing you have to remember is this is the command center. As the command center, you need to take in information everywhere. So the composition of your cerebral spinal fluid, could that affect what your hypothalamus does? Yeah. Could hormone levels in the blood affect your hypothalamus? Yeah. Your hypothalamus is the command center. So while primarily it's neural, our hypothalamus is going to get controlled by everything. It is the starting point. So it collects all information, all data, including top-down information from our nervous system. So it's the combo platter. It gets it from everything. Yes, and because obviously it is incredibly complicated, how is it controlled? Is that a question I'm likely to ask on the exam? Exactly. Nobody knows how the Illuminati functions, so we're not even going to try to guess at it. So yeah, so you can put a big X through that one too. We won't worry about how the hypothalamus is. Although it probably wouldn't be wrong to say neural. If you were going to guess, I would say neural from the central nervous system. So let's put that. I like that. Rather than putting an X through there, let's put neural via the central nervous system. I like that better. But that's our sky is blue answer. But the real truth, let's know that it's going to take information from every source that it can get it. All right. As always, I have done a truly amazing job of drawing this. Again, my artistic skills have just expanded exponentially since I started using the whiteboard and don't have to write in class anymore. But let's take a look at the pretty pictures from your textbook, because they do an okay of doing this as well. Notice here is our anterior pituitary. Notice, again, we have neurons in the hypothalamus releasing those inhibiting and releasing hormones into a capillary, which travels through our pituitary portal, or actually, let's do this. So boxes, releasing and inhibiting hormones into the capillary, traveling down the pituitary or hypophyseal portal system. Remember, those two terms are interchangeable. To our anterior pituitary, where we are going to stimulate the anterior pituitary cells to either make more and release more, or make less and release less of their hormones. And then those hormones enter into the blood. So really there is an exchange taking place in this capillary. The pituitary, the hypothalamus hormones come out, pituitary hormones go in, and then off to the rest of the body. So that is that hormonal control for everything of the anterior pituitary. And for a posterior, neural control. So again, two hormones, antidiuretic hormone and oxytocin, are produced up here, stored here in the pituicytes, and then when the time comes, they fire their action potentials, stimulate the release into the capillaries, and that oxytocin or antidiuretic hormone is released into the blood. So all of the posterior pituitary hormones are controlled via neural control from the hypothalamus.
So we're saying the function of the hypothalamus is that it regulates both pituitary glands?
Yep. Yes, remember, we said the hypothalamus is the command center for the endocrine system. We thought it was the pituitary for hundreds of years. And only recently did we learn that actually, while indeed the pituitary gland controls all the other endocrine glands, the hypothalamus controls the pituitary gland. All right. Excellent. So if we understand this relationship between the hypothalamus and both the anterior and the posterior pituitary, then we can start talking about, how are we on time? Oh, we're doing spectacular. We can talk about some of the hormones of the pituitary gland, the hypophysis. And again, there are half dozen, 8, something like that, 1, 2, 3, 4, 5, 6, 7, 8, 9, something like that that you need to know. 10, I guess, with the melanocyte stimulating. No, melanocyte stimulating hormone is there. So 9, is that what's on your list? I don't have the list in front of me. You guys are really gonna make me bring it up? Here, hold on.
We have 8 and then 9, including the melanocyte.
Okay, well, so that's cool. But we've already done the melanocyte stimulating hormone, so we don't have to do that one again. But yeah, so there you go, perfect, 9. So let's talk about those. We will start first with the posterior pituitary. Again, it is nervous tissue. So remember, what's special about this is the posterior pituitary is the only endocrine gland that does not make its own hormones. These hormones, antidiuretic hormone and oxytocin, are produced solely by the hypothalamus. So the posterior pituitary just stores and releases it. All right, the only one where it's different that way. So let's talk about them, starting with this one first, one we've already talked about, and we'll continue to talk more. Antidiuretic hormone. What is the target of our antidiuretic hormone?
Kidney.
Kidney, excellent. And what does it tell the kidney to do?
Reabsorb water.
Reabsorb water, absolutely, excellent. So if we think about it, there's two main effects of that. It tells the kidney to reabsorb water. Remember, as we talked about, your kidney filters 200 liters of filtrate during the course of the day. And 99% of that is drawn back in. Over 99% of that is drawn back in. But antidiuretic tells it to draw back more. And if it draws back more, that does two things. There is less water in your urine, so your urine becomes concentrated. However, is that the important thing? Do we really care about the concentration of our urine? No, we're drawing that water back into our body, back to our blood volume, so it increases our blood volume, which of course increases our blood pressure. So it brings blood pressure up, brings blood volume up, because it tells the kidney to bring more water back. You might not have thought of it in these terms, but you're aware of it. Because while, how is our antidiuretic hormone regulated?
Neural control via hypothalamus.
There you go, neural control via the hypothalamus. But antidiuretic hormone release can be affected by other things. Caffeine decreases the production and release of antidiuretic hormone. So when you have that quad shot of Dutch Brothers in the morning, right, an hour and a half later, you have to pee because that caffeine is suppressing the antidiuretic hormone. So your kidneys bring back less water. You produce more water in your urine and produce more dilute urine. So your bladder fills up faster with more dilute urine. And what else has that effect as well? Not just caffeine, but what else?
Alcohol.
Alcohol, absolutely. Right after that third Long Island iced tea down at the college bar, right? Then, you know, once you break the seal, that's it. You're going all night long. In fact, that dehydration from suppressing antidiuretic hormone, because you're releasing so much water, is one of the main factors that contributes to the hangover the next day, right? And don't get even started on what happens if you drink Four Locos, right, with the caffeine and the alcohol, right? Then you just become a shriveled up raisin as a result. All right, questions on that? Excellent, love my stunned silences. Let's talk about the second posterior pituitary hormone, oxytocin. What is the target of oxytocin?
Uterus.
Uterus and mammary.
Excellent. So mammary gland is definitely one of them. And what's the other one?
Uterus.
Uterus. Excellent. Right. So it targets the uterus and it targets the mammary glands. What does it do to the uterus?
Causes contractions.
Contractions. This is part of that positive feedback that we talked about. Right. We'll talk about this more when we get to the reproduction system. But when that basketball is ready to come out, it is face down, right? Head down, face down, trying to push its way out. And as the head gets pushed against the cervix, that cervix stretches. That stretch sends a neural signal to the hypothalamus. Hypothalamus sends a neural signal to the posterior pituitary, and the posterior pituitary releases oxytocin, telling the uterus to contract. That pushes baby harder against the cervix, stretching the cervix more, sending a bigger signal to the hypothalamus, a bigger signal to the posterior pituitary, and more oxytocin is released. And more and more and more bigger and bigger, enhancing the stimulus, right? That's what a positive feedback system does, until finally the contraction is so powerful, baby is expulsed, body's back in homeostasis. Life no longer is, but the body's in homeostasis. What effect does it have on the mammary glands?
Milk production.
There you go. Not production, but milk letdown. Absolutely. It allows for the letdown of milk. The mammary glands produce milk because of a different hormone, which we'll talk about in just a moment. But producing the milk isn't enough. To express the milk more easily, what happens is the milk is moved from the glands down into these lactiferous sinuses, these big loose spaces that it's easier for the baby when it's suckling to get that milk to express. Well, there you go. Knock, knock. Knock, knock.
There.
Who's there? Excellent. Pavlov, just checking. All right. Who was Pavlov?
Yeah, dogs.
He had dogs. And what did he do with those dogs?
You rang a bell and gave them food.
Gave him a steak and rang a bell. And when he gave him that steak and rang the bell, they started to associate the bell with the steak. So eventually he could ring that bell and they would salivate, right? Operant learning, right? These learned reflexes that we get. Well, that's exactly what happens with milk letdown, right? Mom produces milk to feed the baby. When baby's hungry, baby cries. So when baby cries, we feed the baby. And so mom starts to associate a baby crying with feeding the baby. So after a first couple months, mom and dad finally get a chance to go out on their first date alone by themselves, away from the newborn baby. They're just happy to be out in the real world, right? Walking through a mall or something like that, and somebody else's baby cries, and what happens?
Mom feels milk coming.
Mom lets down milk, exactly, right? Moms also, when they have to go back to work, take advantage of this. You can look at a picture of your baby or watch a recording or listening to the crying of the baby, and that can actually help with the milk expression if they have to pump. Yes, Ryan, great question. So far, let me forget. All of the hormones released by the hypothalamus and the pituitary gland are amino acid based. Yes, that is correct. Excellent. So again, not milk production, but, well, so there you go, Alex. Notice everything we've talked about is female related, right? So is oxytocin produced by males?
Female.
Yeah, exactly. And Alex has it correct. It is what is often referred to as the intimacy hormone. Now, again, the key is indeed intimacy, right? It is a close personal connection. Doesn't necessarily mean sexual, right? Having sex with someone in a booth in the back of a bar isn't going to increase your oxytocin levels. Intimate contact with a loving partner, right, including sex, can increase oxytocin levels, but so can sitting on a park bench holding hands, right, or cuddling with someone in the bed. Or in fact, they've shown that if you make eye-to-eye contact with your dog, staring into the eye of your dog and having your dog stare back into your eyes elevates both your and the dog's oxytocin levels. It plays an important role in intimacy and bonding, and because of that, oxytocin is sometimes referred to as the cuddle hormone, because it plays that role in the bonding, in the intimacy of the bonds that form between those. Again, we're not talking about cheap, meaningless sex. We are talking about intimate, close relationships. All right. And of course, let's beat the dead horse. How is it regulated?
Neurally by the hypothalamus.
Perfect. Excellent. Questions on that? So what I would say for the cuddle, and again, remember, it's not just males, but females also get that oxytocin elevation as well. My guess is that the target would be nervous system, probably the limbic system. That's where many of our emotions and memories and things like that are. So I would say the target for the intimacy, I would say, is nervous tissue, and then probably the limbic system.
On the picture for the oxytocin for the males is smooth muscle and ductus, deferens, and prostate gland.
Okay, so oxytocin, so both for oxytocin or for prolactin?
Yeah, oxytocin.
Okay, so oxytocin has potentially shown an effect that it may enhance ejaculation. So it may have an effect on that. Like I said, it can be related to that intimacy, but it doesn't elevate it, like I said, in just sex. It's the making love part, the intimacy part of it, that where you see those effects. So I would be fine with those as targets as well. All right. Let's switch to the anterior pituitary, talking first about human growth hormone. What is the targets, plural, of human growth hormone?
Bone, skeletal muscles.
Yeah, many of the cells, most of the cells in the body, but in particular bones, particular cartilage, particular skeletal muscle. And of course, what does it do?
Growth promotion.
Yeah, it promotes two things. It promotes cellular growth, right? But it also increases the metabolism of the cell. In particular, making more proteins, right? Helpful for the muscles in that they grow bigger, they grow stronger as a result of that. And it also helps to encourage the body to metabolize fat instead of carbohydrates. Very powerful stuff, right? We've learned how to take advantage of this. There are, for instance, there's a certain type of dwarfism where what happens is the epiphyseal plates close too quickly. So if we give those individuals higher levels of human growth hormones prior to that, they are able to increase height, in some cases by as much as 3 or 4 inches. Now, I know that doesn't sound like a tremendous amount, but if we're talking about the difference in height between 3'10" and 4'2", are those 4 inches significant? Yeah, absolutely. Of course, people with certain types of dwarfism aren't the only ones who take advantage of this, are they? Who else takes advantage of these?
Athletes.
Athletes, absolutely, all right? Not so infamously, Barry Bonds, local hero, right? Took all sorts of examples of human growth hormones, right? Now, how come he wasn't 10 feet tall because he was taking this? If it makes you taller, why wasn't he taller as a result of it?
His plate was already closed.
Yeah, his epiphyseal plate's already closed. He took this late enough in his career where he had also already mostly finished puberty. His epiphyseal plate's closed, so his bones didn't get longer, but did his muscles get bigger? Did his bones get denser? Did his head get huge as a result of that? Absolutely, of that abuse of these hormones. Absolutely, right? So they take advantage of this a lot, right? But part of the problem with that is it leads to increased ossification of the cartilage. So often people who abuse human growth hormones tend to have joint issues, especially in their hips, especially in their knees. Right? And again, while there are certainly examples of individuals like Barry Bonds, like Mark McGwire, Sammy Sosa, right, who all took advantage of these and all got massive, there's still plenty of examples of athletes who are getting popped in professional sports for human growth hormones. And especially some of these scrawny pitchers you see using them, right? It's kind of a little surprising, right? Because they're not massive, they're not big. And that's because what people found out is you didn't have to be massive. Yes, being really big and strong like Barry Bonds and Mark McGwire made them be able to hit the ball further, but they still needed to be able to hit the ball. Right? So it doesn't necessarily make you a better player. It makes you a stronger player. But what they found is that, again, I know, and again, COVID is a whole different thing, but back in ancient times, baseball players played 162 games over a summer. Right? And again, it is a game, but it is a tremendous amount of grueling wear and tear on the body. And one of the advantages of things like human growth hormones is they can overcome and repair and rehabilitate from injuries and just from the normal wear and tear of playing ball. If, 2 months into the season, your muscles are still strong, your bones are still strong, your body's not wearing down as much, you're going to be able to play longer and play better and still be able to hit the ball. So it wasn't just about getting big, but it's about being able to maintain that high level of activity for a prolonged period of time. So you still, while you don't see the people as massively crazily abusing them like we did in the 90s, there are still plenty of people taking advantage of the resources that the human growth hormones provide. And since this is our first anterior pituitary hormone, how is it regulated?
Hormonal regulation by releasing and inhibiting hormones of the hypothalamus.
Perfect. Hormonally regulated by releasing and inhibiting hormones of the hypothalamus. And yes, you're right, Ryan. It can help to both protect from injury and also to recover more quickly from injury, as well as from recover from wear and tear of just, running hard for 9 straight innings during a baseball game.
Isn't that what they gave that guy, that Netflix documentary, the football player who murdered somebody? They gave that to him so he can keep playing football, and they said that he had brain damage.
I'm not familiar with that particular. I know there are many football players who had been accused of using human growth hormones, but I don't know about the particular Netflix documentary you're talking about. I haven't seen that.
You're talking about Aaron Hernandez?
Oh, yeah. So my understanding is that he has been accused of using human growth hormones and especially like back in college and stuff. But I'm not familiar enough with this case. And from what I understand, he had a lot of other issues going on as well, anger issues and sexual orientation issues and things like that that may have affected that as well. That being said, abuse of hormones can be associated with mood swings that can affect aggression and things along those lines. So yes, so it could have been something related that way. There is a correlation between the abuse of these things and increased aggression as well.
It was like he had gotten too many concussions or something, and it shrank his brain.
Yeah, it was
exactly. CTE is another one that could be a major issue. Yeah, there've been a lot of issues of people who have had to deal with CTE, and so yeah, it's an interesting field. All righty. Any other questions on this? All right. Next anterior pituitary hormone, prolactin. What is our target here?
Mammary glands.
Mammary glands, absolutely. Excellent. And what is the effect on the mammary glands?
Make milk.
Yes, this is the one that actually causes milk to be produced. Excellent, right? Again, is this something that can occur in males?
Maybe.
Maybe. Yeah, it can actually happen. Now, again, males do normally produce this hormone. We'll talk about the effect of it in just a second. However, one of the things that when we're talking about the abuse of steroids and the abuse of human growth hormones and things along those lines, often one of the things that athletes do to mask that is they take feminizing hormones. For instance, when, what was his name? Manny Rivera? No,
yeah.
I'm sorry?
You're talking about boxers?
No, I'm talking about the baseball player.
I know who you're talking about. I don't know.
It's Manny something. Yeah, there you go. Manny Ramirez. Manny Ramirez. Last time he was popped for illegal substances in his body, it was actually estrogens. he was taking estrogens because estrogens and prolactins are one of the things that they would use to try to mask these hormones. And especially, you saw it more in the 80s when steroids first became a major abusing thing, that one of these things would happen is you get these big, huge, massive bodybuilders, you know, with that huge roid rage that people would talk about. And in some extreme cases, they would actually start lactating as a result of this from taking these types of masking hormones. So they could actually cause lactation. Again, you don't see it that much because they've gotten more sophisticated in the cheating, but where you see the use of prolactin today artificially is in adoption situations. When a couple or a female adopts an infant, one of the things that that female can do is take prolactin supplements to actually be able to mature her mammary glands to be able to produce milk. Now, typically it's not enough to fully feed the baby, but it can provide some nourishment. And more importantly, it's used more for bonding with the baby by being able to produce that milk that way. But again, males aren't necessarily producing milk on a regular basis, but it is produced by males, and it seems to play primarily a role in increasing testosterone production. So it may play some role in the maturation or the enhancement of reproductive functions, kind of similar to what we saw with oxytocin. All right. And of course, how is it regulated?
Hormonally via hypothalamus with the releasing and inhibiting hormone.
There you go. Exactly. I think we've beaten the horse enough where I'll stop messing with you guys on it that way. All right. Excellent. Alphabet soup terms. I love my alphabet soup terms. Adrenocorticotropic hormone. Now, the good news is on an essay question, once you've written it out once, you can abbreviate it ACTH. But don't do that on the lab exam. But like normally when we have a big alphabet soup term, it tells you everything about it. Adreno refers to what? Adrenal gland. The adrenal gland, like many things we have talked about in this class, has a chewy nougat center and a candy-coated shell. It is a big triangular gland that sits on top of the kidney. And no, those are not drawn to scale. But the adrenal gland has a chewy nougat center and a candy-coated shell. And when we have this type of orientation, what do we call the inner region?
Medulla.
Adrenal medulla.
The medulla. And what do we call the outer region?
Cortex.
Cortex. So the adrenocortico talks about which part of the adrenal gland do you think?
The whole.
Well, cortico means or refers to the cortex. So this hormone targets the cortex of the adrenal gland, and it is a tropic. Anybody know what tropic refers to? Primarily growth. Tropic refers to growth, stimulation. So it's a big alphabet soup term, but it tells you exactly what it does. It stimulates the cortex of the adrenal gland. So a big alphabet soup term, but it tells you what it is. So of course, the target is the cortex of the adrenal gland. And its effect is to stimulate the production of a specific class of hormones called glucocorticoids. Well, it stimulates the growth, not so much of the size, but stimulating the cells in the cortex to produce glucocorticoids. So it produces a class of hormones called glucocorticoids. And with a name like glucocorticoids, these are lipid-based hormones that play an important role in regulating our glucose metabolism. Not talking about insulin and glucagon. We're not talking about things that way. What we're talking about is mobilizing our glucose to deal with a stress response. Again, from a biological standpoint, right, stress responses were life and death situations where you had to fight that bear with an axe or you had to outrun it. So obviously that required energy. And so when we are in a stressful situation, we need glucose to be able to regulate that. So it plays an important role in helping to regulate our metabolism of glucose to help us to deal with a stress response. All right. Questions on that? All right. And of course, regulated hormonally, releasing and inhibiting hormones of the hypothalamus. Now we once again have a class of hormones. And this class of hormones are called gonadotropins. Guess what gonadotropins affect?
Gonads.
The gonads, there you go, right? And those are the testes or the ovaries, depending on what flavor of person you are, right? There are two specific gonadotropins. One specific gonadotropin is follicle stimulating hormone. And as we already mentioned, it targets the ovaries and the testes. Now, of course, is it going to have the same effect on the ovaries and the testes?
No.
No, of course not. So on the ovaries, it is going to lead to the production of estrogens. Estrogens is, again, a class of hormones. The most specific type, most common specific one, is estradiol. So estrogens are a class of hormones, half dozen plus different hormones, and the most common one is estradiol. And in males, oh, and of course that leads to the maturation of our oocyte, the egg. And in males, it leads to the production of sperm. Notice we didn't say making eggs. Why isn't it making eggs in females? Anybody know?
Females already have all the eggs they're going to have when they are a fetus.
There you go, absolutely. Right, in females, being the overachievers that they are, while they're inside developing in mom, they're already working on the grandkids. Right, a female is born with all of the eggs she is ever going to have. So that's just about maturing the ones that are already there. Whereas males don't start, producing sperm till puberty. Of course, then they don't stop till 15 minutes after they're dead, but so they make up for lost time that way. All right, questions on those two?
Could you give this hormone to men to up their sperm count?
So you could, but here's the problem. And this kind of goes back to what we were talking about with the human growth hormones or androgens or other things along those lines. What is the primary way that hormone levels in our body are maintained?
The hypothalamus.
No, you're right. It is the command center. But remember, how did we say that we regulate the levels of most of the hormones in the body? What is the primary way we do that? Negative feedback. So let's think about this. As a male, I've got a big weekend coming up where I need to impregnate my woman, right? I didn't want to use the term wife, so whatever. So I want that extra boost. So maybe I take a shot of follicle stimulating hormone to increase my sperm production in anticipation of that. Well, now as we'll learn, sperm production takes 90 days, so it wouldn't do me that good to take that now. But again, that's beside the point. The point is, if I had that spike of follicle stimulating hormone, what my body's going to see is, whoa, there is a ton of follicle stimulating hormone in my body. So what is my anterior pituitary going to do? It's going to stop producing it, and it's going to stop releasing it. And so it's going to suppress my normal production and release of follicle stimulating hormone. So then next week, after the effects of that shot have worn off, my body's going to have a hard time bringing the levels back up. And so now my levels are low, and I have to take another shot, and it suppresses it even more, and another shot, and it suppresses it even more. That's what happens with a lot of bodybuilders. When you hear these stories about, mostly you hear about pro wrestlers, because pro wrestlers are much more open and honest about some of the abuses that they took with these things. Many of these older professional wrestlers are now having to take androgen supplements because they were taking so many of these androgens to get big and strong when they were playing as wrestlers, that they have suppressed the ability of their body to produce their own so much, their body no longer produces their own natural androgens. So now they have to take supplements just to lead a normal life. So yeah, so doing that may give you a very short, boost. But as we'll see in the reproductive system, it takes 90 days to make a sperm cell. So again, it's not going to help you for this weekend or anything along those lines. And you run the dramatic risk of dramatically influencing your body's natural processes. Yeah, like all the cells in the body, they will reproduce but again, it's about their ability to be able to produce those. We can suppress the ability to produce those where they're not going to be functional. So you'll have plenty of cells in the anterior pituitary. They just won't necessarily be able to produce it. Yeah, so it would suppress your own ability to produce follicle stimulating hormone. And if you suppressed it enough, it wouldn't come back. Or it wouldn't bounce back to the levels that it was, or it may take years for it to bounce back to the levels that it was. All right. Great questions. Any others? All right, let's talk about our, So theoretically, you've got the right idea. Somewhere there must be a thermostat control that determines what we consider a normal level of follicle stimulating hormone, right? But do we currently know what specific gene that is that does that and how to modify it to increase those levels? Not necessarily. And as we'll see when we get to the reproductive system, there's more than just one implications of many of these hormones. Again, remember, a lot of what everything we're talking about, we could spend an entire semester just talking about the endocrine system. We could talk about an entire semester just talking about the reproductive system. So some of these processes are incredibly elaborate and incredibly complicated. So, theoretically, might it be possible to do something like that? Theoretically, but do we have any idea how we would go about doing that right now? No. And like you said, as we've learned, because of interactions and things like that, there may be many other cascade effects of that. Absolutely. All right. But remember, follicle stimulating hormone is only one of our specific gonadotropins. The second is luteinizing hormone. Still a gonadotropin, so it's still targeting the ovaries and the testes. But this time it has different effects. Luteinizing hormone changes the female's ovary to produce progesterones. Now, again, progesterone is the specific hormone. The class is called progestins, but you'll see why I use the specific hormone here in just a minute. So the class is progestins. Progesterone is the most common specific one. Progesterone is the hormone that tells the female body the egg has been released. Fertilization can happen any minute now. Get ready. So it tells the female body to get ready for possible pregnancy. So it's that big, huge warning bell. In males, luteinizing hormone. Oh, and it triggers ovulation. In males, luteinizing hormone produces testosterone. So notice progesterone, testosterone, those rhyme. So that's why I like doing it that way. Testosterone is, of course, a specific hormone. And, of course, the class is androgens. Androgens is the class of hormones. Testosterone is by far the most common androgen. But androgen and progestins don't rhyme, so that's why I like progesterone and testosterone, because it helps me to remember them. And, of course, testosterone is our mascularizing hormone, and we'll talk more about that later. All right. Questions on that? So, in some, let me say it this way. In most men, as they age, there is a minor decrease in androgen levels, in particular testosterone. However, the changes are relatively small and relatively gradual, so that you have 80-year-old men who are still capable of producing viable offspring, right, with their trophy wives that they're too old and frail to be even able to pick up, right? So while there can be decreases in those, the decreases are not always significant. Now, are there some cases where the decrease can be significant, where they can have some effect, right? It's almost impossible to watch a football game these days without seeing a commercial or two about low testosterone levels, right? Low T values and things like that. Decrease in luteinizing hormone could be one of the cause, but it could also be problems with the cells that produce the androgens as well. So it could be both ways. It could be luteinizing hormone, it could be the cells, and of course, depending on which it would depend on how you treated it, right? If you increase the amount of luteinizing hormone that person had, and the problem is the cells aren't producing as much as they did before, then more luteinizing hormone is not going to make that any better. But if the problem is that you have low levels of luteinizing hormone, then yeah, a luteinizing hormone supplement could increase those levels. All right. Last, but certainly not least, at least I believe this is last, thyroid stimulating hormone. Obviously, what's the target?
Thyroid gland.
Thyroid gland. And what is its effect? What did we say it tells the thyroid gland to do? Yeah, tells the thyroid to produce the thyroid hormones. And specifically T3 and T4. All right, thyroid hormones is the class, T3 and T4 are the specific examples. And just because it's the last one and we want to beat the dead horse, how is it regulated?
Releasing and inhibiting hormones from the hypothalamus.
There you go.
Regulation.
Hormonally via releasing and inhibiting hormones from the hypothalamus. Excellent. And there you go. Just that simply, we have gone through all of the hormones of the anterior pituitary.
So can we write T3 and T4, or do we actually have to write out thyroxine and triiodo something? I don't know how to say that.
Iodothyronine, yes. On the so here's what I would say. If the question was what is the effect of thyroid stimulating hormone, then there I would have no problem with you saying it regulates production and release of thyroid hormones. You could use the class, or you could even say T3 and T4, okay? However, when we talk about the thyroid gland, and I point at a cell in the thyroid gland, and I ask you to identify the specific hormones that are produced there, then you have to spell out triiodothyronine and thyroxine. Okay? See, since here we're kind of indirectly talking about them, it would be okay to talk about them as a class or to even abbreviate them here. But when we are talking about them directly, you need to spell them out. Does that make sense? Or if all else fails, spell it out. If you've got to learn it for the gland itself, then you might as well use it here every place you can. Besides, saying triiodothyronine is a lot of fun.
How do you spell that? TRIIODOTHYRONINE? Is that right?
Oh, I didn't put it on here. Yep. So, okay, I don't have it there, but we'll get to that in the next class. There you go. Triiodothyronine. So, someone asked about melanocyte stimulating hormone, then it has a star next to it. Someone remind me again why it has a star? What does the asterisk indicate?
It's made in the pars intermedia, and it's not always made.
Excellent. It's made in the pars intermedia. And yes, it's not that it's not always made, but it is typically not a hormone that we have circulating in our body.
Potentially.
Yeah, it's a hormone that can be potentially made there, but in a typical, normal, healthy individual, we don't produce it. However, if something caused us to produce it, it would target the melanocytes and increase melanin production. So we do know what it would do when it's produced, but it's not something we normally produce. And because it's not something we normally produce, do we have to worry about how it's regulated? No, you do need to know its function. You need to know its target. You need to know its function. Right? Targets melanocytes, stimulates melanin production. But what you don't need to do is worry about how it's regulated. So that was the one X you've gotten to put on the graph. Everything else we filled out. All righty. That's everything. That's all we're going to do for today. Questions on any of that? So, starting Thursday, we're going to do the rest of this. We are going to look at the histology. We are going to talk about the classes and the specific hormones, targets, effects, how it's regulated, and we're going to finish off the rest of that chart as we work our way through the rest of the organs of the endocrine system.
You have to know that, like, cortisol as a specific type of, or a specific type of glucocorticoid.
Yes. So typically, so here's what I would say. Typically, anytime you need to know a, oops, why is this not working? Typically, anytime, well, that's weird. I don't know why it's not letting me draw on this. All right. Weird. I don't know what's going on here. Okay. Typically, anytime you need to know, oh, I know why, because I'm not sharing it anymore. That's what I want. There we go. Typically, anytime you need to know a class of hormones, you need to know typically the most common. Specific example. So for androgens, the most common specific is testosterone. For estrogens, the most common is estradiol. For gonadotropins, you need to know both specific hormones, follicle stimulating hormone and luteinizing hormone. And as we're going to start, the next class is for our thyroid hormones, the class, you need to know both specific hormones, triiodothyronine and thyroxine. So yes, you need to note at least one, if not more than one, for any of the classes that we will talk about. All right. Any other questions on this material before we call it a day? All right, we survived the first rough endocrine day. The second day will be a little bit better. We'll probably finish a little bit earlier than this, which means we should have time for a question and answer review. So please have questions on any of the material. Do not forget about the lymphatic system. Do not forget about the immune response. Those are on our exam as well. I know between all the stuff we're covering on the endocrine and the fact that you're working on your projects, it's easy to forget about the first part of this section. But I would say that this exam is pretty evenly split between half of it being lymphatic and immune response and half of it being endocrine, both on the lab and the lecture exams. Lab exam might be a little bit more endocrine because there's a little bit more anatomy there and a little bit more histology. A lot of histology on this test. Get used to looking at your slides. All right. Which reminds me of one more thing. I have not posted it yet, but over the weekend, I'll write this here. Is this still recording? Excellent. I will write this here. I will try to find the link and I will add it to, if I can find it, I will add it to our website. I'll add it as like an interesting link. It isn't something that is necessarily going to be, let me rephrase that. It is not anything that is going to be on the exam. But since we were talking about our immune response and we were talking about COVID-19 and we were talking about vaccines, there is a podcast by the name of Sawbones. Sawbones is an interesting biology podcast. It is a married couple. The male is basically a podcaster, comedian guy, jokester, but his wife is a practicing physician, MD. And basically what they do is they talk a lot about the history of medicine, right? All of, like leeching and other things like that, common ancestral myths and things that, when we talked about the humors of the body as the four humors, being, bile and all these types of interesting things. And they talk about, they talk about interesting topics. It's somewhat amusing. They did one on vaccines, very recently one on vaccines. They've done a couple on vaccines, but the one recently, here, see if I can find the episode number. They've had a lot of COVID ones recently. Shucks, hold on. There it is. It's called Operation Warp Speed. Operation Warp Speed from July 24th was what the name of, and again, they got rid of that name pretty quickly, how they were going to rush out the vaccines for COVID. And so it's episode 333. The only downside about it is that it was during one of their, I guess there's a couple weeks where they are doing a big push for donations and things along those lines. So there was a couple more advertisements in it where they want you to sign on for money and stuff like that. But it was very interesting. They talked about how they're forming these vaccines, how the vaccine formation is different, the difference between DNA and RNA proteins. Remember, we were talking about whether you can have antigens or whether it's the weak or the dead. It was very, very interesting. It related a lot to what we were talking about. And so I think it is definitely something that, like I said, it isn't necessarily information that's on the test, but as you're trying to make yourself knowledgeable and aware of what's going on in the world, I think it was something that was really worthwhile to listen to. They asked a lot of other really interesting ones, some fun ones, some interesting ones, some scary ones. And again, by scary, I mean just, the things they talk about that people have done and are doing to their body. Again, it's family friendly, so they don't curse. They keep it's not too graphic or anything along those lines. So it's highly interesting. It's an entertaining podcast, but this one in particular, I think went really well with what we were talking about. So if you're interested, check that out. All right. Any other questions or anything else? All right. That's all I have for you for today. I will see you. Rest up for Thursday. Yes, and get your popcorn ready for the presidential debate. Absolutely. It should be entertaining. All right. Take care, guys, and I will see you on Thursday.